Okay, good morning, ladies and gentlemen. Thank you for having me. My name is Sonia Nanta, college student of the chemistry department, Riau University, and I'm going to be your host in the first session of this international webinar with the title Green Approaches in Medicines, Chemicals, and Energy, in conjunction with the 58th anniversary of chemistry department, Faculty of Mathematics and Natural Sciences, and Riau University. The Honorable Director of Riau University, Professor Aras Mulyadi, or the representative, the Honorable Dean of Faculty of Mathematics and Natural Sciences, Dr. Samsuduha, Master of Science, or the representative, and the Honorable, our speakers, Dr. Taifo Mahmud from Oregon State University, United States of America, and Dr. Muhammad Asif Khan, PhD from Perdana University, Malaysia, and Basmi Alam Fakih University, Turkey. Ladies and gentlemen, before we start our webinar, please stand up for singing the national anthem of Indonesia, Indonesia Raya. Thank you. Please take a seat again. Okay, this is the next schedule. I will explain about the schedule of this first session that will be appear on your screen. So this is the schedule of session 1. Okay, we will move to the next event of the session. The opening remarks that will be given by Director of Riau University or the representative to the representative of Rector Riau University. Time is yours.
Baik, Assalamualaikum warahmatullahi wabarakatuh. Syukur alhamdulillahirabbil alamin. Pada pagi hari ini kita dapat bertemu dalam suatu event webinar internasional yang ditaja oleh jurusan Kimia Fakultas MIPA Universitas Riau dalam rangka berkaitan dengan Milad ke-58 Universitas Riau. The Excellencies, Rektor Universitas Riau, Profesor Dr. Insinyur Aras Mulyadi, The Excellencies, Dean Faculty of Mathematics and Natural Sciences, and as well the head of study program Chemistry, Faculty of MIPA, and the Excellencies, keynote speakers, participants, and all of us. The Excellencies, Profesor Dr. Taiful Mahmud. Selamat gabung, Prof.
Terima kasih, Pak.
Very late night in your area, isn't it, Prof? Sorry, Prof. Because very good morning in Indonesia. Good morning, Indonesia. Still in morning, Indonesia. This almost late night in your area.
It is 7 o'clock.
Professor Taif. Okay, thank you, Prof. Professor Taiful Mahmud from Oregon State University, USA, will be delivered a speech regarding to approach in medical chemistry. And the second speaker in morning session is Muhammad Asif Khan, PhD, from Perdana University, Malaysia, and Besnialem Fakif University, Turkey. He will deliver entitled Bioinformatics, a key driver of a green approach in medicine. And an afternoon session, there are two speakers, Dr. Raihana Begum from Energy Research Institute, Nanyang Technology University of Singapore, will deliver entitled about colloidal semiconducting quantum dots from synthesis to application. And a second speaker, Professor Dr. Eric Hirsch from University of Groningen, Netherlands, will deliver speech entitled Green Energy Harvesting from Biomass. Peserta webinar yang kami hormati, participants from all over the world. I have noticed Mrs. Saini from Thailand, Mr. Owen Tylers, Peter Kier, Lara Alfred, and others. As well, very respected our college from participants all over the nation, from the western part of Indonesia, Unsia, until the eastern part of Indonesia, University of Samratulangi. Welcome all of you in this seminar. This seminar entitled Green Approach in Medicine, Chemicals, and Energy is conferred by Jurusan Kimia MIPA Fakultas MIPA Universitas Riau in conjunction with the 58th University of Riau, 1st of Oktober 2002. Peserta webinar yang kami hormati, kami sebagai penyelenggara berharap kepada para peserta, Bapak Ibu, terutama yang lebih khusus adalah kolega muda kami, para dosen muda, para peneliti muda. Semoga kehadiran dalam webinar ini akan dapat menggali pengalaman dan dari presentasi-presentasi yang disampaikan untuk memperkuat kapasitas Bapak, Ibu, dan saudara dan teman sejawat dalam bidang riset yang ditekuni, sehingga akan meningkatkan kapasitas. Dengan lebih khusus, pada pagi hari ini akan disampaikan berkaitan dengan kajian medicine, chemical, and energy oleh para narasumber. Selain dari itu, kami berharap wahana ini juga sekaligus merupakan kesempatan untuk membangun jejaring atau link dengan teman sejawat atau senior-senior researcher kita di bidang yang terkait untuk meningkatkan kualitas riset ke depan. Secara ringkas dapat kita sampaikan bahwa objektif dari webinar ini diantaranya adalah gaining an experience upgrading the capacity building, and creating a networking. Mudah-mudahan dengan waktu yang sangat terbatas, dengan event yang sangat penting ini, Bapak, Ibu, saudara, kolega, rekan-rekan dosen, dan para dosen muda, dan para peneliti muda akan mendapatkan pengalaman yang tidak ternilai. Dan akhirnya dengan mengucapkan basmalah, Bismillahirrahmanirrahim, webinar ini kita buka secara resmi. Akhirnya, terima kasih kepada seluruh narasumber dan panitia. Selamat berseminar. Kami akhiri dengan 2 bait pantun. Bunga lembayung kembang setaman, mentari bersinar di ufuk berawan. Selamat bergabung encik, tuan-tuan dan puan-puan pada webinar jurusan Teknik Kimia FMIPA dalam rangka Milad Universitas Riau ke-58. Mari kita sukseskan. Asal sembilu dari buluh dianyam menjadi tempian. Kami susun jari yang 10, atas khilaf dan salah mohon dimaafkan. Billahi taufik wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, thank you for the opening remark that has been given by the Rector of Riau University or the representative. Okay, we will move to the next event, but before we enter the next event, I will introduce about the rules in this webinar. So let's check it out. Okay, that's all about the rules. Okay, without any further ado, let us give the stage to our moderator, Dr. Rudy Hendra. To Dr. Rudy Hendra, the stage is yours.
Thank you, the host. Good morning and Assalamualaikum warahmatullahi wabarakatuh. I hope all is well. Thank you for the committee for having me here. My name is Rudy Hendra. I am an academic in Department of Chemistry, the University of Riau, and I would be your moderator this morning. Welcome to the Green Approaches in Medicines, Chemicals, and Energy webinar organized by Department of Chemistry and the University of Riau. Thank you to all the honorary guests and the speakers, as well as all participants to attend this webinar from Indonesia and outside Indonesia. The participants can ask a question by writing down in the chat room during the presentation in English or Bahasa, and the committee would translate it in English. So do not worry if the participants are not able to write down in English, we will help you to translate it in English. And then for the first lecture would be delivered by Professor Dr. Taiful Mahmud for about 45 minutes. Before we start our first lecture, I would like to introduce you Professor Taiful's academic background. Today, Professor Dr. Taiful Mahmud will deliver a presentation with the topics Green Approaches in Medicinal Chemistry. He is a professor in College of Pharmacy, Oregon State University, USA. He graduated his PhD from Osaka University of Japan. He attended postdoctoral at University of Michigan and USA, and then as a research assistant professor in University of Washington, and then co-founder and vice president of Garusol Laboratories. And his research expertise are natural products, biosynthesis, genetic engineering, and he has been awarded for many grants and awards. So thank you, Professor Taiwo, for your time to give a lecture this morning or evening in Pacific time. I believe it's about 7-ish p.m. in the evening. So without further ado, please welcome Professor Taiwo, and the floor is yours.
All right, thank you very much for the kind introduction. Can you hear me?
Yes, you're
Yes.
welcome.
Oh.
We are very clear.
So first of all, I would like to thank the organizer for inviting me to give a talk in this webinar. And I also would like to congratulate University of Riau and the Department of Chemistry for the 58th anniversary. And so today I would like to talk about green approaches in medicinal chemistry. And actually it has been a pleasure really to be able to attend or to join this webinar. And I have seen a few familiar faces like Professor Titania, and also surprisingly, I saw Dr. Shaini and Dr. Pakit from Thailand also in the audience. So both of them were in Corvallis, Oregon State University, doing research as well. And I know one of your faculty members, Dr. Yuana Nurilita, well, she spent 6 months here in my lab last year. So it feel like, talking to a friendly crowd. So it's really nice. Anyway, so this picture really show Oregon State University during fall term, like right now, and it's really nice with the leaf, color change and so on. But several months ago, like 2 months ago, so we had a pretty bad wildfire here in the West Coast, on the West Coast of United States. California, Portland, Oregon, and Seattle, a lot of wildfire. It was unprecedented. It's very big. And since I was here, I've been here for 23 years now. And that was the first time I saw really big fire very close to cities. And of course, sounds quite familiar with people in Indonesia as well, like in Riau, from time to time, you have wildfire as well. So apparently this is an indication of global warming and the environmental problem that we have more recently getting worse and worse. And obviously everybody need to do their part here to preserve this world and the globe, and then try to apply kind of green way or environmentally friendly ways in our daily life. As a chemist, of course, we need to do the same. So green approaches for chemistry have been developed or have been used for quite some time now, and then many people develop green solvents using, instead of using organic solvent from petrochemicals, they try to develop, chemical reaction using water, for example, or extraction using supercritical carbon dioxide, and so on and so on. And then, of course, we can also develop green catalysts or use green catalysts like biocatalysts, enzyme. Of course, many people have been using, lipase or acyl or esterase enzyme or protease and so on. And then another possibility also using green and renewable raw materials like natural products, right? And then green methodologies as well, microwave induced chemistry, for example. And for people who are working in the biotechnology area or natural product biosynthesis, for example, using genetic engineering or synthetic biology to generate new compounds instead of doing organic synthesis or total synthesis from scratch. And also, of course, in silico drug discovery using computers and programs to help us or to guide us to find candidates compounds instead of randomly synthesizing many different compounds. So in my lab, basically we have been working on studying a number of these, like biocatalysts. We have been working with enzymes and trying to find new enzyme as new catalysts. And then also my background really natural product chemistry, and I've been working with natural products for many, many years now. And we also use genetic engineering, synthetic biology in silico drug discovery as well. So in the next 40 minutes, perhaps I would like to talk to you or to introduce some of the projects that we have been working on for a number of years now, as an example of these green approaches to medicinal chemistry. So the first one is rifampicin. Rifampicin is a first-line anti-TB drugs. As many of you know, that there are 4 anti-TB drugs that's considered as a first-line anti-TB drug, including isoniazid, rifampicin, pyrazinamide, and ethambutol, right? And rifampicin is a semi-synthetic compounds derived from rifamycin B, which is a natural product. This is a polyketide natural product. So it's a very complex compound. It's produced by a soil bacterium called Amycolatopsis mediterranea. And this compound kills bacteria by inhibiting bacterial RNA polymerase. So these 4 hydroxy group here are very important for the binding to the enzyme RNA polymerase. So rifampicin has been the #1 anti-TB drugs because it's very active. And also isoniazid, for example, and also very cheap. So it has been used for a long time. And although it requires a long time to treat TB patient, requires 6 months to treat them. So basically, we have very good anti-TB drugs so far. But unfortunately, the incidence of TB, tuberculosis in the world is still very high. In 2016, it's very recently, so there were 10.4 million people were infected by TB. And then among them, 1.3 million people died because of TB. So that's very, very significant number. And perhaps is the #1 killer among infectious diseases that have drugs to treat them. And but more recently, even worse, because there are multi-drug resistant TB or rifampicin resistant TB as well. So over time, the organism become resistant to this very good drug otherwise. So in 2016, there were 600,000 people were diagnosed with multi-drug resistant TB. And then about 240,000 people died because of that. Unfortunately, Indonesia is one of the countries that has a very high incidence of multi-drug resistant TB. So in fact, Indonesia is one of the 30 MDR TB high burden countries. So in this case, oh, this one move. In this case, Indonesia is not only, have a lot of multi-drug resistant TB, but also incident of TB and HIV. So of course, when people are infected with HIV, the virus, and then over time they will develop, AIDS. And nowadays, of course, there are many good drugs to treat HIV infection, and many people with HIV infection can live normally. But in more rural areas or in certain countries, of course, many people, because of the the price of the medication, maybe they didn't get enough or they didn't get the medication. So over time, their immune system become lower and lower, and then they get infected by TB. So the TB HIV combination is a deadly combination, really. And then, of course, the number of TB in Indonesia also very high. So this is really concerning. So why bacteria gain resistance toward rifampicin? Apparently, over time, bacteria develop mutation in their RNA polymerase enzyme. So in the wild type of RNA polymerase, so rifampicin can fit very well into the pocket and block the production of RNAs because it's RNA polymerase, right? But there are several mutations can occur with RNA polymerase. For example, D516, this is aspartic acid, can mutate to valine, and then histidine can mutate to tyrosine, and serine can mutate to leucine. So in this case, the IC50 can change very dramatically, especially in this mutation. The wild type, the IC50 of wild type is about 0.012 micromolar, and suddenly change to 1,130 micromolar. So the bacteria are completely resistant to rifampicin. And the frequency is much lower compared to the serine mutation. It's very high frequency, but the effect to the rifampicin a little bit lower. So that's the problem with rifampicin-resistant Mycobacterium tuberculosis. So in collaboration with my colleague here in the chemistry department, Professor Ha-Young Chung, we calculated the free energy of binding of rifampicin to the mutant. This is a resistant RNA polymerase. And we compare that with this hypothetical structure, 24-desmethyl rifampicin. So we thought a methyl group at this position. And by this in silico calculation, we found that this hypothetical structure actually has a better binding affinity to the mutated RNA polymerase. So, of course, we talk about this, well, it would be nice if we can get this compound, but the problem is how are we going to get this compound? Because this compound is not produced by the bacteria. Just missing one methyl group at that position, it is also almost impossible to remove one methyl group from rifampicin to get this molecule. We look at the literature, many people did total synthesis of natural products, apparently there were not that many people have synthesized rifamycin. And what we found was rifamycin S has been synthesized by Professor Kissy from Harvard University in 1981, and then rifamycin W. This is even not active actually against bacteria. It was synthesized by Professor Kuniaki Tatsuya's lab and was published in 1990. And looking at the synthetic scheme, it is very long synthetic scheme, and the yield, the overall yield was very low. So it is almost impossible to make the compounds that we want using total synthesis. And of course, it is not a green way to do it as well. So we thought about using nature or natural machineries to make this compound. So we look at the biosynthetic machinery of rifamycin in the producing strains, Amyclotopsis mediterraneans, and what happened there? And so the bacteria has been sequenced, the genome of the bacteria, and then we know that the bacteria contains the biosynthetic gene cluster of rifamycin. This is a polyketide synthase gene, and then also we have the HBA biosynthetic gene and many regulatory genes in the cluster. As many of you know, genes are a blueprint in organism, and gene can be used as template to make RNA, messenger RNA, and from messenger RNA, you can make proteins, in this case, enzyme that can catalyze the production of rifamycin. So there are a lot of different enzymes involved, and this has been done, the study has been done by Floss, Hutchinson, Shoup, many different groups, including our groups. And so we know that this compound, this complex molecule is synthesized through a very complex or very interesting machineries. So started from this small molecule called 3-amino-5-hydroxybenzoic acid as a starter unit. And then this compound is extended one by one. So 3 carbon unit, and then 2 carbon unit, and 3 carbon unit, and so on, to make this long chain polyketide compounds. And then RIF-F here is the amide synthase that can close this ring to make a cyclic molecule. And then of course there are many regulatory or tailoring genes or enzyme that can modify the molecule to make the final products. So this polyketide synthase machinery has been studied quite well so far, during the past 20 years. So a lot of information, a lot of knowledge are available for this system. And then we thought, if we want to make the molecule without one methyl group, right? So maybe we can modify this biosynthetic machinery. So apparently, actually we know that this extender unit or extension of 3 carbon unit is regulated by a acyltransferase domain of the enzyme system. So this acyltransferase domain recognizes methylmalonyl-CoA as substrate to introduce this 3 carbon unit. The next one also 3 carbon unit. The next one also 3 carbon unit. But if you can change this acyltransferase domain with another one that can only introduce 2 carbon unit like this, so presumably we will get a molecule without a methyl group at that position. So basically we go back to the blueprint and then change the blueprint and change the machinery to make the compound that we want. Just an analogy, if you think about a factory making a car, right? You have assembly lines, but if you don't like the lights, you can go back and then change the lights. Well, you can go to the factory, ask them to change the light, for example, right? So so we did this in collaboration with Professor Rup Lal in Delhi, University of Delhi, and then we got the mutant. We call it acyltransferase 6 mutant, and then we grow the mutant, we culture them, and then we screen them, and we analyze the products and compare to the wild type. So the wild type produce rifamycin B and then produce rifamycin SV. Those are natural products that have been known for many, many years. And then our mutant apparently produce a couple of compounds with 14 atom mass unit different. So that's equal or similar to one methyl group missing in our compounds. Of course, we don't know which methyl group is missing. So this is the NMR, proton NMR spectrum of, or spectra of rifamycin B. This is the natural product. And this is the product that we got from our mutant 24-desmethyl rifamycin B. And as you can see, rifamycin B has four methyl groups here, which is consistent with this 4 methyl group, whereas our compound has only 3 methyl group, right? So this is presumably one of them is missing. And then the fact that the shift of this signal quite significant, so that's also indicate that the whole conformation of the compound has been changed or has changed. And to make the rifampicin analogs, as you know, even rifampicin itself is a semi-synthetic product. So to make rifampicin analog, then we have to use some chemical reaction really to convert 24-desmethyl rifamycin B to 24-desmethyl rifampicin. But during this conversion, we found this 24-desmethyl rifamycin S, and through X-ray crystallography, we were able to confirm that indeed the methyl group at this position is missing. So this is a dimer of this molecule. So with that, we were able to synthesize the compound that we want using biosynthetic pathway really to make the intermediate, and then use a very few organic reaction to make the final product. So this is something that we thought really nice. And of course, you are curious about the activity of this compound. So we tested this compound against sensitive strain of Mycobacterium tuberculosis, and then the resistant strain of Mycobacterium tuberculosis. And then we used rifampicin as a standard. Of course, rifampicin is active against the sensitive strain, but it's not active against the resistant strains. And the compound that we generated basically active against all of them. So this is something that really significant. All right, so the second story that I would like to share with you is about pactamycin. This is another unique molecule. It's very complex, isolated from the soil bacterium Streptomyces pactam. It is a very active compound, active against bacteria, active against fungi, kill malarial parasite, but also kill human cells. Basically, it's very toxic and no selectivity. So it is not a good compound to be developed as a drug. It inhibits bacterial growth or human cell growth by inhibiting the ribosome of the cell. So in bacteria, specifically bind to the 30S ribosomal subunits of the ribosome. And it is not very big structure, but if you are a chemist, you should acknowledge that this core unit is very condensed, and with all these stereo centers, it's very difficult to synthesize this chemically. And it was discovered by Upton Company, in 1960s, and they were trying to develop this compound, but because of the complexity of the molecule and the toxicity, finally they didn't really pursue this further. But this compound has very good biological activities. And after 50 years, after discovered, and finally, Hanesian and co-workers published the first total synthesis of pactamycin in Angewandte Chemi, required 32 linear steps from threonine with about less than 3% overall yield. And then 2 years later, Johnson and co-workers published the same total synthesis in Science, but with 15 linear steps from acetylacetone with 1.9% overall yield. So it's an amazing, very talented group of people that were able to synthesize these very complex molecules. And of course, this opened up the possibility of making analogs or derivatives of the compound for further biological testing. But in our case, we try to avoid using organic synthesis to synthesize the compound from scratch. So we have been looking at the biosynthesis of pactamycin. As I mentioned, the molecule is very interesting. And this 5-member ring here actually is derived from a sugar molecule, the N-acetylglucosamine. And then this compound, the 3-aminoacetophenone, is derived from 3-aminobenzoic acid. And more interestingly, there are a lot of modifications that are needed to make this molecule. And then this modification is very unique because you are making a CC bond here using, well, based on the information that we got from the genes that are involved in making this compound. This reaction occurs through radical SAM-dependent enzyme. So this CC bond formation is unique, right? And of course, then by looking at this cluster, we know that there are a lot of interesting enzyme in that system. So that's really encouraged us to study this system more deeply. And indeed, we found a lot of interesting biocatalysis involved in this reaction. And then of course, we clone the genes and then we produce the enzyme one by one and then carefully characterize this stepwise reaction. And what we found very interesting is that this reaction involves a polyketide-like enzyme using this acyl carrier protein. So we start with acyl carrier protein PTMI, and then it's being activated by this phosphopentadienyl arm. So this is an arm that can take the substrate, put it in the end of the arms. And then this ketosynthase enzyme then catalyze the coupling reaction of these two substrates through a Claisen condensation to make this products. And then this compound is then glycosylated by PTMJ to make this product. So as you know, glycosyl transferase or glycosylation occur all the time and is a very common reaction in nature. But glycosylation of a small molecule, while it is still attached to a protein, is very unique and unprecedented. So if you're interested in this, you can see our paper that was published last year here in Nature Chemical Biology. And then more recently, we also discovered, even more interestingly, by studying more enzyme downstream, and this sugar is further modified while still attached to the protein. So from alcohol to ketone, and then from ketone to amine, and then this amine being carbohylated to make this molecule. So, this is a difficult experiment to do because we have to use, protein mass spec to analyze all these products. And then further downstream of the pathway also involve a number of very interesting tailoring processes. For example, like I showed just now, this is a CC bond formation. Is catalyzed by PTMH. This is a radical SAM-dependent enzyme. And then this is the N-methyltransferase enzyme that methylate this amino group to this dimethylamine. And then PTMR is a Cas3-like enzyme that is responsible for the attachment of 6-methyl-salicylic acid compound to this primary alcohol to make 7-deoxypactamycin. And then this PTMY is a cytochrome P450 enzyme that introduces a hydroxy group of the final products. And many of these enzymes, actually, they are very promiscuous, meaning they are flexible. They can use different substrate that give us a benefit of using this enzyme as biocatalyst. Anyway, by knocking out the genes and making mutants, and by engineering the biosynthetic pathway, we were able to make a number of compounds, derivative or analogs of pactamycin. You can imagine if we have to synthesize this molecule synthetically from scratch one by one, that could take many, many years. And then also we can introduce fluorine, for example, by feeding fluorinated substrate to the bacteria. So assuming this is a bacteria, you have a lot of scheme or pathway to secondary metabolism, then you can block the pathway, for example, by knocking out genes. You can make it to detour to something else. So this is a very powerful tool or powerful method to make analogs of complex natural products such as this. As I mentioned just now, some of the enzymes, they are very flexible. So one of them is PTMR. This is a Cas3-like enzyme. Cas3 is a ketoacyl synthase enzyme that usually involved in making fatty acid or making polyketide compounds. But in this case, this enzyme function as an ester transferase or acyl transferase. So basically transferring this 6-methyl-salicylic acid from a iterative polyketide synthase enzyme, PTMQ, directly to this primary alcohol, and you get the final products. So as I mentioned just now, this acyl carrier protein usually has a phosphopantatenyl arm that attach the substrate here. Of course, if we want to do this in the lab, in the test tube, it's more difficult to make this kind of substrate. So what we did, actually, we make a SNAC, N-acetylcysteamine derivative like this, just to mimic, to resemble this part of the molecule. And luckily, the enzyme is able to take this compound as substrate as well to make the final product. More interestingly, we also can change the substrate or the acyl with many different type of acyl compound like this. And we were able to make derivatives or analogs of pactamycin. So you can see we were able to make libraries of compound like this using enzymatic approach or chemoenzymatic approach. So right now we only produce in very small scale, and we are trying to scale up this reaction, and then we will do structure activity relationship study to look for active compounds. And then if you look at the analogs of pactamycin, some of them I just show you here. If you compare this compound that we produce through genetic engineering of the strain with the parent compound pactamycin, as I mentioned just now, pactamycin has very strong antibacterial activity, strong antimalarial activity, also very toxic, right? And just by changing this functional group from hydroxymethyl here or ethyl to methyl group only here, it's completely changed the biological activity of the structure. Now, we found the compound has no antibacterial activity, but still retain antimalarial activity, almost the same IC50. And then about 30 times less toxic compared to the parent compound. So this is an amazing thing to see how natural products, the small change of the molecule completely change the pharmacological properties of the compounds. So that's something, you know, we need to be very careful about when we compare or when we discover something which is slightly different from the compounds that has been known. You cannot just assume that they have similar biological properties. There could be completely different biological properties. This is the last part of my talk. Let me see. Okay, so for many years now, we have been working also on a group of natural products that we call C7N aminocyclatols, or we call also pseudosugars. Because this type of molecule contain compound or structure that looks like sugar, but it is not sugar. One example is Acarbose. This is an anti-diabetic drugs produced by Bayer. This molecule resembles oligosaccharides, but because of the structure of this compound and the CN bond here, this become an inhibitor of alpha-glucosidase enzyme in the stomach. So that's the reason why this compound is being used as anti-diabetes. Another example is validamycin A. This is another similar compound contain pseudosugar or not real sugar molecule. And validamycin is being used as an antifungal agent, but specifically to treat crops, to treat rice plant. So every year, it's about 30 to 50,000 tons is produced in China, and is being used in Asia, in Vietnam, Thailand, and many other countries to treat fungal infection in rice. Of course, rice is one of the main source of carbohydrates, and we, in Asia, we eat a lot of rice. So by having this type of molecule that can, can be used as crop protectant, really play a significant role in maintaining healthy rice production and food security. So this is the molecule of validamycin A. As you can see, it's contained structure that looks like sugar, but they are not sugar. So there is no oxygen in the ring. So as I mentioned just now, it's being used as antifungal agent to treat sheet blight disease in rice plant. And this disease is caused by Rhizoctonia solani. And Rhizoctonia solani is a fungus that use trehalose, this is 2 units of glucose, as energy storage. Whenever necessary, they would hydrolyze this trehalose by the enzyme called trehalase to make 2 molecules of glucose through this intermediate called oxocarbenium transition states. And validamycin A actually in the cell will be hydrolyzed to validoxylamine A. As you can see here, validoxylamine A looks really similar to this oxocarbenium transition state structurally. So this compound actually can bind to the enzyme trehalase. And because of the CN bond here, it cannot be hydrolyzed by the enzyme. So basically inhibit the activity of trehalase so that fungus cannot get glucose and they cannot grow or they will die. So for many years now, actually we have been working on this project. Of course, we are very interested in knowing how this unique sugar-like molecule is made in bacteria. So we have, studied this step by step very tediously from the beginning, apparently, in fact, it is produced originally from pseudoheptylose 7-phosphate, which is the intermediate of the pentose phosphate pathway. And then this sugar phosphate is cyclized to make a cyclic molecule like this, and then further modified, and then you get these two different intermediate, and then these two different intermediate then coupled together to get this validoxylamine A, 7-phosphate actually, and then it's then converted to validamycin A. And then of course, during this study, we have published quite a bit of report on this. And during this study, we discovered a number of very interesting enzyme or biocatalysts that we thought, require or need further investigation. So one of them is 2-epi, 5-epi baliolone synthase here that is responsible to cyclize this sugar phosphate to make this cyclic molecule. And then the other one is what we call pseudoglycosyl transferase enzyme that catalyze the coupling reaction between these two non-sugar compounds to make validoxylamine A. So I would like to discuss a little bit about this enzyme. The first one, so we obtained the X-ray crystal structure of the enzyme. And this enzyme looks very similar to dehydroquinone synthase, which is involved in the cyclimate pathway in many bacteria and plants. This type of enzyme that we call sugar phosphate cyclase enzyme is very unique because this enzyme can catalyze 5 different reactions, all occur in the same active pocket. So if you look at this pseudoheptylose 7-phosphate here, the first one is an oxidation at this position to make a ketone. And then dephosphorylation reaction occur to make this intermediate. And then reduction of the ketone back to the alcohol. And then ring opening, and then the rotation along the C5-C6 bond to make this intermediate. And then this is intramolecular aldol cyclization. So 5 different reactions all occur in the same enzyme. So this is very interesting. That's the reason why you can get this cyclic molecule from the sugar phosphate. And then there are a lot of studies that we have done with this enzyme, but because of time, I just want to quickly show you where we have been, we are going with this. So we can use this information to mine the genome database and find other possible sugar phosphate cyclases in nature that nobody know their function. So recently of course we have been doing this for years, but recently we look at 35,680 unique protein sequence and then align them and using bioinformatic approach, study them and then differentiate or group them based on their potential function. And well, actually in total there are 6 new enzymes, 7 with the dehydroquinone synthase. And this enzyme actually was discovered by somebody else, but we have been working on these 4 enzymes. And the EEVS or 2-epi 5-epi baliolone synthase that I introduced to you just now is here. And then now we have this enzyme that we call it new enzyme because we still don't know the function of this enzyme. So currently we are still working on this. And the other enzyme also, have been studied in our lab for quite some time now. And then the second enzyme is called pseudoglycosyl transferase enzyme. The reason this is unique because this catalyzes this coupling reaction of 2 non-sugar molecules. But it's resemble glycosyl transferase enzyme. So looks like glycosyl transferase, but instead of sugar, you get non or pseudo sugar molecule to make this compound. And then currently we are working on the catalytic mechanism of this reaction. And I think the mechanism is different from regular glycosyl transferase enzyme because there is no, this is not a glycosidic bond. So the reaction can be different. And in fact, well, the question was, are pseudoglycosyl transferase ubiquitous in nature? Can you find other pseudoglycosyl transferase? As many of you know, glycosyl transferase enzyme is very ubiquitous in nature. There are more than 105 families of glycosyl transferase enzyme because a lot of natural product being glycosylated, you get glycolipid, you get glycoproteins, you get many compound contain sugar. They are all catalyzed by different families of glycosyl transferases. And through our study, we have studied, well, we found a number of possible or putative pseudoglycosyl transferase enzymes that are similar to glycosyl transferase enzymes. So, we also separate them based on their families. So hopefully one day we will able to establish a new superfamily of enzymes that are similar to glycosyl transferase enzyme. So I think my time is up. So in summary, as I've shown you, I hope I can assure you that green chemistry really has become increasingly important in an era of global peril from environmental problems and climate change. So obviously every one of us need to think about how we operate, and how we do our work and then in our daily life as well to be more environmentally friendly. And I've shown to you that biocatalysis can play an important role in green chemistry for chemists. Of course, it's something that we may need to look into. And natural products have served as green and renewable raw materials in medicinal chemistry because they can be, produced relatively easily if you know how. And then, of course, you don't require a lot of organic reagent and so on. And then I've shown to you as well by synthetic studies of natural product can lead to the discovery of new compound as well as new enzyme or biocatalyst. And then many different approaches that you can use like biosynthetic engineering, chemoenzymatic approach, or synthetic biology. We have several other synthetic biology projects that I don't have time to talk about. And all these things really provide new ways or new avenue to make new drugs. And then they are powerful tools really for us to be able to make complex molecules. So with that, I would like to thank the people who really did the work, my past and present group member, and then a lot of collaborators and funding agency that fund our research. So this is one of the group gathering, and actually this is Dr. Yuana Nurulita from University of Riau. Last year we had barbecue in my backyard before COVID-19 came to visit us. With that, I would like to thank all of you for your kind attention, and I'm happy to answer any question. Thank you.
Thank you very much, Professor Taiwo, for the nice and comprehensive presentation. Okay, for the next part, it's going to be a Q&A session. Before, I think so for the warming up, I'm going to have one question for Professor Taiwo. Well, it was good, nice slide, especially in the first slide, your picture in the autumn leaves. Okay, I want to ask you about the pectomycin, isn't it? Is it correct names? Yeah.
Pectomycin.
Yeah. As you mentioned, is in the structure there are more than two stereocenter. Yeah. So I do believe that you figure out the orientation for the structure by using CD spectroscopy. So because I mean last year I have a chance working with SIO bromotyrosine with Ted Molinsky in UCSD. So I just figure out just one stereocenter, but what should we do if we have a more than one stereocenter by using CD spectroscopy?
Right. So, well, I know Ted doing a lot of CD experiment, but it's not every compound is appropriate with CD. So, and of course, in many cases, you can use NMR data first to identify the relative stereo configuration of your structures. And then after that, you can use many different approach, like, using MTPA ester, for example, or you can use CD if you have a chromophore that you can use. And then of course, nowadays, ECD, you can calculate the possibilities of different configuration. Yeah. So, but the one that like to use really in the past, requires some kind of functionality that he can, put something on it and then run the CD. But nowadays, with ECD also, and in this case though, the structure is really complex. You have 5 different stereo center within that 5-member ring compounds. And, but again, you can get the structure, the relative stereo configuration just by NMR. But then you just then have to identify one of the functional group for absolute configuration, and everything is fine.
So instead of using the NMR and then the calculated coupling constant, this is enough for to determine the stereocenter for this type, but with the complex stereocenter structure.
Not correct. So the coupling constant, calculated coupling constant only can be done if you have enough protons, on this 5-member ring, but you don't have that many protons that you can calculate the coupling constant. So, but through NOE, for example, Roisy, for example, then you can get the relative stereo configuration. Relative can be flipped. So that's the reason why you still need to use CD or something else, or MTPA ester to characterize the absolute configuration.
Sorry, I mean that's NOE or Roisy. So thank you, Professor Taifo, for the answer. Okay, for the next questions, maybe I will start with Professor Titania Nugroho from Chemistry Department, University of Riau. I think I will read the question. Thank you for Professor Taifo for every interesting lecture about the promiscuous enzymes. A well-known promiscuous enzyme is the oxidoreductase enzyme lactase. Have you ever used a lactase to try modifying one of the compound that you work on?
No, I have not done. I have not used lactase to modify the compounds.
Oh, okay. So you haven't.
But we use oxidoreductase quite often.
Okay, thank you, Prof. I hope that's enough for Professor Titania Nugroho question. Okay, the next question. Oh, I think she has a two question. The second one is TMO26 derivative from pectamycin. Was it obtained by using the enzyme to modify it, or did you use a gene knockout strain?
Right. So originally we produced TM26 by gene knockout. So, but we can also make TM26 using chemoenzymatic reaction. So we can convert TM25 to TM26 using enzyme. So we can make the compound using both methods.
Okay, thank you. Okay, we go to the next question is from Lucy Purba from University of Riau. Thank you, Professor Taifo, for the insightful lecture. I want to ask about the feasibility of your research. What do you think about the cost analysis of your research? Would it going to be more accessible than the current antibiotics in the market? It should be a lot.
So this is very very interesting question. That very important question, really.
Yes.
So well, we take it the way how I think about this is two different aspects. One is from a scientific standpoint. So what we are doing, being in academia, so one of the role is really to produce knowledge. So to produce knowledge is an important part of our work, right? So, and not too much about the economic calculation of that, but from doing basic research like this, you never know, what you get, and then sometimes they are very, very important. And then, all this, the big finding or all the stuff that you enjoy right now, or the compounds that are produced by company, they all come from basic research, most of them. And then in our case, from the research that we are doing, we have been doing, and then some of them have been, has led to the intellectual properties, and then even lead to a spin-off company or biotech company that use our technology to make products. So yeah, so it's interesting question, but well, it is an important question.
Yes, thank you. It looks not important, but it's very important because it's about the money. Okay, the next question is, can you go to the next, change the next slide? I can read the question.
It's enough for the session, sir.
Oh, okay, thank you.
There is no more question.
Okay, is there any more question from the participant? Can raise a hand, direct question. No more question. If there is no more question, thank you very much again for Professor Taifo Mahmud for your comprehensive presentation. I hope you enjoy your days and then enjoying the autumn leaf. I think in Corvallis, or maybe it's already fall. And have a good night. And thank you very much on behalf of the committee.
Thank you.
Thank you. Goodbye. Okay, for the next speaker, for the next lectures, it's about bioinformatics, a key driver of green approaches in medicine, where will be delivered by Dr. Muhammad Asif Khan. And beforehand, our second lecture, I would to introduce Dr. Muhammad Asif Khan academic background. Dr. Muhammad Asif Khan, he is academic from Perdana University School of Data Science, Malaysia, as well as Besmialim Wakif University, Turkey. He got his PhD degree from National University of Singapore. where he finished his postdoctoral research fellow in National University of Singapore in 2010, and then as a visiting research in Johns Hopkins University, USA, and then in Osaka University, and then also he is EXCO board member at Global Organization for Bioinformatic Learnings, Education, and Training until now. And he also, as a president of Asia Pacific Bioinformatics Network, or AP Bionet. His expertise are bioinformatics, data warehousing for biology, and vaccine informatics 1. So without further ado, please welcome Dr. Muhammad Ashrif Khan, and the floor is yours.
All right. Thank you, Dr. Rudy Hendra, for the kind introduction. Let me just
You're welcome.
share my screen. Okay. All right. So, sorry. Okay. Sharing screen. Okay. All right. Can you see my screen?
Yes. Thank you.
Okay. Perfect. All right. Thank you so much for that introduction again. It's a great pleasure to be able to join for the international webinar today. And I just want to say that I'm right now in Turkey in my kitchen, keeping quiet a little because my kids and my wife are all sleeping. I didn't realize when I agreed to this that this will be 5 in the morning here. Well, it's 6 now. So you might see, this is my kitchen, so you might see the sun coming up in a bit. All right, so I'm gonna speak a little quieter. I hope you all can still hear me. Am I audible well?
Yes.
Are you able to hear me? Okay.
Yeah, I can.
Perfect. Okay, great. So yeah, I'm in Istanbul, Turkey, and I'm also affiliated with Perdana University Malaysia. I'm actually with Perdana University Malaysia. I'm over here as a visiting professor for 3 years. There's a fellowship and a long story, but yeah. So I'm with both organizations. So my title, you're aware of my title. I just want to put a disclaimer out there. I don't claim to be an expert in green approaches. So, but I tried my best to put in as much as possible. I'm sure it's not that comprehensive, but I hope it gives you some picture of what is being done in terms of making medicine more green. And of course, my focus will be on bioinformatics. So yeah, let's get started. So I just want to congratulate the chemistry department and Riau University on their 58th anniversary. That's a huge deal. You never, hopefully we look forward to the 100th year anniversary. And this is a big deal. So congratulations for coming this far and continuing on the road to glory. And I hope Riau University will be a light for knowledge for the wider part of Indonesia and the region. So once again, hearty congratulations from my side. And my next slide, I just wanted to showcase that I have been to Riau University, and this is actually some sort of pictures, a number of pictures, walking down the memory lane. Back in 2017, there was a workshop organized by Professor Bimo Tejo and Prof. Collis. This was at the university, and I was invited to be one of the workshop speakers. So here I am. And I have my flight ticket there. So as a proof that I did buy and I did go there. This is back in the days when, we didn't know about COVID, the good old days. We could just fly easily. It's a bit difficult now. Hopefully things will get better. And one of my memories, notable memories from that visit is this durian pancake. So there's a picture of a durian pancake there. I really enjoyed them. And I think I even bought quite some to bring back. So yes, and that's the group that was there. My colleague Farhan is also there. So thank you again for remembering me and inviting me to give a talk today on this occasion, special occasion. Okay, so moving forward, while preparing for this, I was just trying to see, okay, what would be a proper definition of green approaches? Maybe that's something we should share with the audience. So I'm sure all of you know what it means, but just for formality's sake, okay, so what do we mean by green approaches? So there might be a better definition out there, but this is what I found: environmentally friendly processes, or eco-friendly, nature-friendly, or green, approaches. Loosely speaking, you could call that too. Basically, are sustainability and marketing terms referring to goods and services, laws, guidelines, and policies that claim, I think this is the key part, claim reduced, minimal, or no harm. No harm is a little difficult often, but at least reduce or minimal or no harm impact on the ecosystem or the environment. So that's going to be the essence of what I'm going to talk about, how bioinformatics or data science in the bigger light is actually helping move towards that direction. And there was a nice paper that I came across. I put the link over there that actually had some notable key points that I wanted to share with the audience. So understanding how the world works and how we can do things better. That's, I think, one of the key goals. That's why we do research. We do research to understand the world around us and how things functioning together to bring about the outcome that we are seeing here. But I think the key is to be able to perform research, carry out research more effectively, more efficiently, and of course more safely. That's something that we should always strive towards. And we should, while we are doing that, we should aim to reduce waste of any sort of resource. And this is not just an important reason for doing research, it's also an important part of the process of research. This should become integrated as part of the process. It should not be an afterthought. So when we are designing our experiment, when we are putting that grant proposal, we should think of some of these elements and how we can best integrate them, or what we are doing to do this. If many of you have watched the Apple keynotes, they usually at the end, showcase check marks on how whatever they're doing is contributing to a green earth. So something like that would be nice if we could also put in our funding grant proposals or our own research experimental designs. What are we doing to make it more green? And so we should try to cut down as much as possible. So research itself should not, therefore, not be done wastefully. We should not just say, I got all the money in the world, or I got a lot of money. It doesn't matter. Just buy and just do what we think we should be doing. We got to have that mental switch that we should try to save as much as possible, even if that means not utilizing the funds fully. And no matter how important we think our results might be, we should not lose sight of this focus. So essentially what this means is we should bring about the maximum knowledge and understanding with the least possible investment of resource. And why? Because we are living in a resource constrained world, and this is important for environmental and social sustainability of the planet and of course the ecosystem. And therefore, not just doing research and thinking about reducing wastage and increasing value, we should also have research that tries to figure out ways that we can achieve this. So that is also something that's welcomed. Essentially, research that helps us understand how we can reduce the footprints of trials. Research is all about trials. So if we can reduce that footprint, I think that is something that is also much needed, maybe paramount before even starting the full-on research, so that we can bootstrap it as much as possible and maximize that outcome. And this is where actually bioinformatics comes in, this last point here. It is aiming towards reducing that footprint of trials, reducing that search space. This is actually a term that we use. And if you have a full circle, and that's 100% of the search that you'll be expanding upon, exploring upon, in order to do your research, can we use bioinformatics or data science to cut down that search space and maximize your output, not just output, but also chance of success. So, sorry, it's Azan, it's Subo time. So I'll just continue. I hope that's okay, and you hopefully can still hear me.
I still can hear you.
Okay, great. Thank you. What we have to do just to accommodate, and I hope that's okay. Okay, thank you. So this is another thing that I came across. This was actually a nice article from The Lancet. This is a summary of that article. I provided the link, so any of you interested can read more about this. And what's interesting is, when we think about wastage, we usually think of electricity consumption, buying materials, reducing the materials, but it's not just that. Actually, it's really built into the design of how we carry out research. And this was quite interesting. Sometimes we don't even think about to that point. For example, even the publications that we publish, are we making them green enough, for us to cut down wastage down the line? So this is something that I highly recommend reading. So very quickly, what are these 4 stages of wastage in research? So the questions really relevant, and of course this is in the context of clinicians and patients. So at the question level, when we're asking the questions, how relevant these questions to clinicians and patients. So we have, of course, low priority questions. How high priority questions? Are we asking the high priority questions, or are we asking the low priority ones? Of course, we should be putting in more effort on the high priority ones. So prioritization is something that's worth considering. Are we assessing the outcomes of that research? For example, we are doing this research, but we are not involving the clinicians, we are not involving patients and other stakeholders. These are just some examples of things that are being done. That's wastage. And similarly, our design and the methods, accessibility of our publication. Is our publication fully accessible? Can people get hold of it? Can people read it in time? In order to cut down repeat of experiments that's already carried out. This is definitely critical. We don't want to repeat what has been done. It's kind of a pure wastage. And of course, unbiased and usable reports. I don't want to go into details. I think this is somewhat self-explanatory. You can read more about it. The key is increasing value and reducing wastage. We want to overall increase the value of whatever we are doing. And I like this term that I've came across, knowledge translation. We use the word knowledge discovery a lot. So KT was some sort of something new for me. And I plan to use it a lot. So knowledge translation, the process of closing the research to use gap can provide, closing the research gap can provide solution to reducing research wastage. So thinking about the translation, the end outcome. And of course, something that was highlighted is the missing link, which is probably the funding system. A lot of this probably could be curbed if the funding agencies themselves give importance to this aspect, and maybe that top-down approach could have a larger impact rather than bottom-up approach. And another number or a figure that I found quite astonishing. 85% of research waste is equivalent to over $100 billion dollars per year. That's a huge number. Imagine how many research we could fund with that amount of money that just wasted. So again, the missing link, the funding agencies. So if we could integrate all this thinking and bring all the stakeholders together, then we could definitely push in this direction of reducing wastage, and maybe having more money for more research to be carried out. All right, so moving forward, and I also felt we cannot talk about green approaches without touching on United Nations Sustainable Development Goals. Many of you may have seen this or may have not seen this, but there are like 17 different goals that are being pursued by United Nations to make our planet Earth more a better home for everybody. The green approaches for research specifically contribute on SDG 3. So the short form is SDG, Sustainable Development Goals. So SDG 3 would be of direct relevance here. I think the other one would be probably SDG 12, Responsible Consumption and Production. So this could also impact on the materials that we use, like energy efficient computing or the equipments that are being used in the lab. They could be more energy efficient, so there could be some impact there. And so these are the direct ones that I can see. I'm sure some of the other ones or maybe directly or indirectly impacted by having more green approaches, implemented for medical research. All right, so so that was a little on green approaches. So I've covered one aspect of my talk. So the other aspect, the other keyword of my talk is bioinformatics. So let's learn a little bit about bioinformatics and see what it is all about. Some of you may be very familiar with this, so I hope you still find something new from what I'm going to cover. It's again, as I covered the definition for green approaches, I think likewise it's important that we also understand the definition of bioinformatics. Bioinformatics, there are actually many definitions out there. If you Google for it, this is my favorite one. I call this operational definition. It's the development and application of techniques from computer science, mathematics, and statistics to address biological problems. In this case, we could say medical problems or life science related problems. It doesn't matter, they all are more or less the same. So bioinformatics is essentially integrating all these different approaches to solve a problem, make it more efficient, reduce that number of trials, reduce that search space. That's ultimately the goal. It should be an integral part of that research process. More and more people have started realizing the importance of this and are integrating this in their research work. There's also philosophical definitions, but I don't want to spend more time on that, since this is not really a talk on bioinformatics per se. So I think this operational definition is good enough. This is basically those people who do bioinformatics. This is what they would describe what they're doing. Just to recap, okay, how bioinformatics has changed things in the past. This is an example of the past. We would carry out experiment, we will get results, and we will publish them. All right, and the present now, some of you may not to criticize, but some people actually still doing this past approaches, but more and more, of course, are doing this present approach, where they do experiment, of course, they get results, they publish, and they put those results in a database. When they have sufficient data, they will put it up in database, and some of them go out and actually publish these databases, or even if it's not published, it's out there for people to take advantage of. So this is the present. And the emerging trend is no longer an emerging. These are old slides, as you can see, from 2005 from my supervisor. The emerging trend actually is probably the current trend now, which is you have an experiment, you get results, you publish it, it goes into database, and then you extract the data from the databases. You use in silico or bioinformatics tools to analyze the data, and then you use that to proposed experiments. So the data, these in silico tools for taking data from the databases, help you in the design of your experiment. So it's more targeted. It's much reduce trials, and then you do your experiment, you get results, and there's this loop where the data goes back and then benefits other people in the future, because as we get more and more data, higher quality data, of course the tools will get better and better. And there's a term that we use, data warehousing. So essentially this whole thing is called data warehousing, which is a big part of bioinformatics, this provides the framework for this process. But bioinformatics itself, you could kind of say there are two types of bioinformaticians out there, or the approaches they utilize. Ad hoc, it's like you just want to solve a problem in a one-time way. You have a problem, you just want to solve it, and that's it. You want to forget about it. So that's the ad hoc bioinformatics approach. It's very focused on just solving a problem without thinking the long-term, implications of, optimizing that solution so that other people can benefit from it. And this is also some sort of, a green approach, which is kind of, not emphasized in this ad hoc approach. So there's another one which I'm going to cover in a bit that's more greener compared to this one. So this is less green bioinformatics approach. So you have a biological system, you do your computational analysis, you make your biological interpretation, and that's about it. Sometimes, this is the perfect strategy because you just want to get something out of it and move on. So then this is completely fine if that's the case. But sometimes you spend so much effort and you don't really, think about the long-term impact of that. So what do I mean by the long-term impact? So the long-term impact is essentially like you have a student in your lab who's doing bioinformatics research and he's just solving that problem, and then the student moves on, and then the next person comes. The next person cannot utilize all the work that was done by the previous student because they just know documentation. There's no the codes or the tools are built very specific to that particular data set. They're not generic enough to be utilized for other purposes. So the person has to kind of start all over. So there's a lot of waste of time, maybe waste of resources, waste of money there. So if the person doing this bioinformatics thinks about these aspects, then the design could be implemented from the beginning in that context, so that the next person coming in is gonna be able to just jumpstart and get on with the research work without having the need to figure out from scratch where things are and how to carry out things, how to do things, especially when there's no overlap between let's say manpower. If there's an overlap, of course it's not as bad, but if there's no overlap, then you could end up wasting a lot of time just to get get started. And I mean, that's just one example. The other example is, when you do these kind of things, you can then, turn the crank and publish more, because you have basically created an ecosystem where you can quickly apply these solutions and solve those problems. For example, if you build a database now, it's going to be easy for the next research question to be asked from the data. You just extract the data and ask the next question and the next question and so on. But if that database is not made publicly available, or even in your own lab, you don't make it privately available, then people have to just start all over and waste so much time in that. And actually, this happens all the time. And one of the reasons why people don't do this and they prefer the ad hoc, because it's faster. You quickly get your results. We often don't have patience. We just want to quickly solve the problem and move on. And then little do we realize that eventually we might come back to that and reuse it. But so we miss out opportunities. So I highly recommend this. If you're doing bioinformatics, you should become more systematic in your approach to applying bioinformatics strategies to solving biological problems. So if you have a biological system, you need to formally describe it. So there's a formal description, so the next person is able to understand, and you yourself, 2-3 months down the road, will not forget what you have done. You have a way to come back to, and of course, you try to mathematically define the problem so that it's much more generic and much more applicable, rather than ad hoc or heuristic solutions. You try to make it more optimal. Think about in terms of proposing more optimal solutions. Unless it's not possible, then that's fine. And then you bring in the existing tools. If there are no existing tools, you create your own tools. And sometimes you have to convert results from different data sets or from different tools and integrate those. So that's also something that you want to think about. And you also want to think about automation while you're doing this, so that we can minimize the manual work in carrying out these things. And of course, largely the biological interpretation. So the blues are still there, in relevance in relation to the ad hoc approach, but it's these additional things that we need to bring into the approach in order to make it more green or more more reusable, and leading to more effective research down the line, and actually more value. You create more value. Sometimes you don't realize that the data we have, we could publish 10 papers, we can ask 10 research questions, but sometimes our focus is just this, and we ask that, and we get it over, and we move on. And and you put your data out there, and then it's the other guys who actually extract the other value. Of course, I highly encourage putting the data out there. It doesn't matter whether other people get the value from that, and it's not us. But if we also think like that, then we can also extract that value, together with other people extracting value from whatever data that we put out there. So a data warehouse is a database in a form that enables extraction, synthesis, and complex analysis of data, which of course supports decision-making and extraction of new knowledge. So knowledge discovery from database is the process of automated extraction of useful information. So you can then, take advantage of that data to ask all kinds of additional research questions. So automation is also one of the aspects that needs to be thought about when you're designing these things. For example, a lot of people, they know BLAST, they will go and do BLAST search, but you can also do local BLAST, and you can actually automate BLAST locally and make it more efficient, more effective. And you can then ask more research questions and don't feel that you're limited from the BLAST on NCBI's website because you're not able to fully control that. So this is just small examples. Now all this is becoming more and more important because we must recognize that data is really growing out of hand. We have data explosion. There's too much data out there. I'm going to give you some more examples. And there are too many databases out there that provide this data. So we have explosion of not just the data, but also databases that provide the data. There's some statistics somewhere that we have more than 2,000 databases that are dealing with biological data out there. So again, I'm not going to go into the detail, but data explosion is one of the key drivers for bioinformatics. And when you have this, of course, data quality becomes a problem. And this is one of the things that bioinformaticians spend a lot of time working on. Sometimes, they jokingly call us data janitors because we spend more time analyzing, more time cleaning and less time analyzing, because the analysis part is the fun part. And usually you run the tool, you get results, and you make interpretations. But getting to that stage is where a lot of time is spent. So even bioinformatics itself has its own wastages that could be cut down. Although it's trying to cut down the wet lab wastages, it itself has its own wastages that can be cut down. And if we apply these systematic approaches, then we can drastically reduce. And of course, these systematic approaches, will help improve our data quality integration from various sources. If we do it ad hoc, then it's going to be much harder for the next person. But if we do it systematically, then we really cut down all the effort that we put in improving data quality. So there's a lot of basically problem here with the raw data: inconsistent, inaccurate, redundant, irrelevant, incomplete, incorrect. And when you look at this, you might be scared to even use the biological data out there. But there is a lot of value there, but we have to do it properly, systematically, so we can really extract the gold from all that data out there that's relevant to our research. This is not meant to scare you, but really to encourage you to make use of the databases out there, but be prepared to spend some time taking care of the data, cleaning the data, processing the data, so you really get the maximum output from that. So I talked about integration. There are various approaches that bioinformaticians apply in integrating data. So again, I'm not going to go into the details of this, but yeah, there's a lot of data disparity that we need to resolve. So if you are a bioinformatician and you're working with patient data, actually right now we are working on COVID-19 patient data, and I can tell you there's just a lot of problem in this data. It's so much so that we even have a data where one person's name with 2 passport numbers. That's just impossible. So we even have that kind of data. And when data is manually input, then the problems are even more abundant. So, but you have to fix these problems before you proceed to look at the value that you can extract from the data. So sometimes different structures, the data come in different formats, different presentations, the search is different. They are available in different locations, and you need to bring them together. Maybe the tools that they use in order to present the data or extract the data are also different. So these are some of the challenges that bioinformaticians look into. All right, somebody's drawing something. You must be bored. Okay, so database creation, and of course the last one of the final outcomes would be to create this database and of course maintain it. So when you create the database, you must make sure that other people can extract the data for their own research. You don't create it, but do not allow people to extract the data from it. You must make it easy for people to be able to extract the data. So you use all kinds of tools to make that happen. And also you need to update the database. You don't just leave it there and it becomes obsolete. Obsolescence of databases is a big problem. And sometimes, students move on and the database doesn't get maintained. So it's important that we maintain this so that the value can also be maintained. No point, putting in 2 years and creating a database, and then after that, it just doesn't get maintained. Unless, of course, you have no choice, and then there's not much we can do. But the other thing is like accessibility of the data. You put in a lot of time and you put it out there, and maybe your funding ran out. The management decided to just cancel remove the support for your compute, and then you just don't have the you just lost the ability to present your work to the public domain. So you could actually find other places where you could dump or provide your inventions or your databases, so other people could take advantage of that. So nature of databases, short lifespan, hard to maintain. This is a real problem. This is also something that bioinformaticians need to think about so that we can maintain that value. Sometimes, a lot of money gets spent. I actually have witnessed in my own supervisor's lab, a lot of time, money gets spent, effort, create a database, and then policy changes, the database is gone. Nobody can access it. The paper is there, but the database is gone. All right, so how can we cut down also those kind of wastages? It's also something we need to think about. Now, there are actually some novel solutions to take care of that. And if you're a bioinformatician, you should explore that. And you can Google for those solutions. So in a nutshell, data warehousing goals are really meant to make all these databases sustainable. So value is also sustainable from this, and these are the various steps. If anybody is creating a database, they should really follow these steps and not follow the ad hoc approach, but follow the more systematic approach. I'm not going to go into the details of this. There are many papers on this. You can read on how to make, implement a data warehousing approach for your own data or databases that you're planning to build. So coming back to bioinformatics. So bioinformatics, I've already given you the definition. I've gone through one aspect of bioinformatics, which is the data warehousing. So the biology, medicine, all that this is where the wet lab comes in, the data, and then they are stored in a database. And this needs to be done in a proper way. You don't just dump it. You've got to follow a process. And I've gone through that process of how to create the databases in the best way possible. Then, of course, data mining happens. You have your algorithm, your tools that will mine on this data and then present that output so that interpretation can take place. So in a nutshell, bioinformatics is about data warehousing, and the other half is data mining. But we spend a lot of time on data warehousing and usually very little time on data mining because it's usually very fast. And so bioinformatics analysis is at the core of all this: algorithm, visualization, biology, databases. The core of this is analysis. This is where the software come in. In a nutshell, what we have is data, and what we really want is a very nice story explaining what the data is all about. And this is already happening. Bioinformatics is really making it possible, and not just biology in general, but even in medicine, this is already happening. So in a nutshell, bioinformatics actually underpins integrative biology, integrating biology or life science that is not just driving medicine, but science, life science in general also, to handle complexity of the biological data from 1 dimension to 2 dimension, even 3-dimensional structures, all the way to 6 dimensions. So bioinformatics is actually working at all these different dimensions to maximize knowledge extraction from the data. So in a nutshell, bioinformatics, I think what I'm trying to highlight here is that it really underpins a lot of research. It's a core aspect of, and one of the first steps that needs to be carried out in biomedical research. Right, so now data in medicine, moving towards medicine now, there are just actually a lot of sources where we can get data. We have the medical records, we can get it from retrospective or prospective research, our own collection, or publicly or freely available data sets. There actually are tons of them there. And of course, commercialized data sets that you got to pay in order to get the data. So these are in general 4 different ways you can get data. Of course, if you run a hospital, you have a lot of those special medical records. But if you don't have access to these, you can always get publicly available data. There are tons of them. So there's no excuse to not having data to do biomedical research, and bioinformatics really makes it very easy for you to get hold of these data. There are just a lot of these databases out there. You can read more about them. I just wanted to talk a little bit more about data. Genomics is slated to be one of the key drivers of this data, big data. You may have heard of probably the term big data. So genomics is one of the big drivers. At the moment, not so much. We have like about maybe less than 10 petabytes per year. But in the near future, by 2025, it's projected that it's going to be the largest contributor of big data to the tune of 2,000 to 40,000 petabytes per year. In comparison, Twitter is about just 1 to 17 petabytes per year, astronomy 1,000, YouTube 1 to 2,000. So genomics is really going to be a big driver, and there are many reasons why this is happening. And this is just genomics. And if you think about healthcare data, which is beyond genomics, it's going to be even bigger, contributor of big data. So medicine really holds, really holds a lot of data, and that's also a gold mine, and with golden opportunities to extract value from that data, knowledge extraction, knowledge discovery, knowledge translation. So those individuals who are in this field really, up for an exciting time. Just to give you an idea on the data scale, so you probably have heard of megabytes, that's 10 to the power of 6 gigabyte. Some of you may still be having gigabytes of hard disk with gigabyte storage, but many of you are probably already on the terabyte scale. Your laptops might be 1 terabyte, portable disks 2 terabytes, so it's becoming the norm. So the next level is petabytes, 10 to the power of 15, but it doesn't stop there. We already have people talking about exabyte storage, 10 to the power of 18, and it doesn't stop there. Who knows, down the line, we might be looking at healthcare data, genomic data, moving into the scale of zettabyte, or the highest that that's the highest terminology. The terminology for the highest storage that's being used at the moment is yottabytes, 10 to the power of 24, and it might go even higher. There might be newer terms if we have really if we push the envelope and we go beyond. So I think at the moment we are quite comfortable with petabyte, but exabyte is becoming a reality. I think supercomputers are already dealing at the exabyte level. So we have 2 more levels to break the barrier, and then who knows, there might be something new out there. So this is really exciting. That's how much data that's out there, and a lot of it is publicly available. And so coming back to genomics, which is one of the key, I'm sure many of you know why this is happening, but just to give you a quick reflection, back in 2001, in 2000, the genomic, Human Genome Project completion was announced. The draft completion was announced. At that time, of 2001, we are looking at the cost of genomic sequencing, 100 million. That's very expensive, but the projection is that with the reduction in cost and advances in the technology, we are looking forward to next year the cost dropping to $100. So when it drops to $100, it's essentially a commodity. Anybody can buy. Anybody can request to sequence their genome. Imagine you wanting to sequence your genome and want to know more about yourself. So you will be able to do that at a very low cost. And when the cost becomes low, we don't think much about it. I always joke that we have iPhones of 3,000 ringgit in Malaysia, or 1,000 Sing dollars, and a lot of it have iPhones, and we don't think many times when we want to buy that. So imagine if sequencing goes down to that level, more and more of us would be wanting to sequence. So this is called personal genomics. So personal genomics is going to be one of the big drivers of creating more data. And how long does it take to sequence this? So these are just these are actually old numbers. So 2013, this was prediction back then, but we are currently, so a more reasonable estimate would be based on last year, whole genome exome sequencing, not the whole genome, but exome sequencing alone, just all the exomes, about 5 minutes. So we could do that within 5 minutes. I think within a day, you can do your whole genome sequencing. So you don't even have to wait the many years that, the Human Genome Project had to take in order to just give us the whole genome sequence. So you guys, we all can now sequence at very low cost and very quickly get the output, and it just creates the research opportunities are tremendous here. And this was one of the reasons why NIH US during Obama administration launched this precision medicine initiative. So it's defined as an emerging approach for disease treatment and prevention that takes into account individual variability, which is your genes, environment, and one thing we often forget is the lifestyle. You can have the best genes, best environment, but if you're a couch potato, then probably you're going to be impacted negatively. So this is a paradigm shift, precision medicine, and this is a nice picture that explains that. So yesterday was symptom-based. You look at the symptom, today is pattern-based, tomorrow will be algorithm-based, but tomorrow is already here. We are already seeing people applying algorithm-based approaches in making decisions and trying to treat people, cure people, or manage patients. All right, so now precision medicine, why is it important? Because we might look very similar, but we're also very complex at the same time. This is, of course, an obvious example where these two organisms have the same number of genes, but they look very different. They're also very complex in different ways. Let me just see, how am I doing with time? Am I still okay with time? Can somebody just give me a heads up?
Yes.
Okay.
You still have
Okay.
a time and about 15 minutes.
Okay, perfect.
Yeah.
Thank you. So here's an example, like, well, they look very different, obviously, and the number of cells are tremendously different, but believe it or not, they have the same number of genes, roughly equal number of genes, but the complexities are there for us to see. And here, of course, these people also look very different, but the genomes, some people, I think it's 99.9% identical. So we are percentage-wise so identical, yet we look so different, even among humans. So there is complexity, at the genetic level that's making us different. So where is this complexity coming from? So forget about the environment; that's a whole different thing. But at the gene level, and we have data for that, and 2000, the year 2000, the human genome draft completion was announced. 2004, I think they got the complete one. 20 years later or 19 years later, we are still far away from curing cancer and curing so many chronic diseases. So we of course have improved our understanding, but we are still very far away from maximizing on that data. We have a lot of data, and data is increasing very rapidly. But nonetheless, we're making progress, and we will get there slowly but surely. So precision medicine, it's an important goal in trying to understand these differences, what makes us different, what makes us different in terms of responding to disease or exhibiting diseases. So understanding these differences is going to be key, and precision medicine is slated precisely addressing that aspect. And just to give you another example, that the gene is important, but the number alone is not the key. What's really happening with the genome, that's where the key is, not so much just how big is your genome. So this is a very nice example. Humans, we have 3 billion bases, but a grass, your typical grass that we don't pay attention so much. Of course, the wheat we do, but they have so much more. They have much larger genome, 16 billion bases, and guess what? Our friendly amoeba, single cell, has 670 billion bases. All right, that's way beyond more than what we have. So just the sheer size of the genome just doesn't explain why we are so different, why we are so complex. All right, so I hope this drives home that message. So where human variation, where does it really come from? This is also an interesting example. This is like, you see huge difference, and it turns out that, this could be a result of just, few changes in our genome. So much so that I have read, like, even one change can result in this kind of differences. So every little difference between the genome is going to be key in understanding what makes us different. And we in general differ from each other, although we might be like 99%, beyond 99% similar to each other. We do have these differences. These differences might be very small. The percentage is so small, but when you multiply to 3 billion bases, you do get a good number of differences. So much so that it's estimated to be about one difference at about 1.2 K base pairs. So in total, this will be few million differences in total if you look at the whole genome. So we do actually have quite a huge number of differences between us, even though percentage-wise it's just so small. All right, so so there's a lot that needs to be studied, and bioinformatics is of course a key to making sense of all that data that precision medicine is interested in. So the generation of complexity is of course there because of a modest increase in gene number. Having a bigger gene number does contribute to that, but that's not the full story. The more sophisticated regulation of your gene expression, transcription, alternative splicing, post-transcriptional regulation, post-translational regulation. So all these actually are the more important, reasons why we are different from each other. Of course, you also have other aspects like microRNA that are becoming quite important in understanding the complexity between people. So these are some of the more key areas. So we got the data, but how the data, how the data tells us about this regulation, we're still far away from that. But we, again, as I said, we're making good advances on that front too. So this slide is to highlight that for us to really maximize, to really push precision medicine to the next level, we really need to have a systematic approach to medicine. And which is, I think, nicely summarized by this slide. So, for example, data, we need to integrate the various data sources. So when a patient comes in, we need to do this multidimensional molecular profiling. So you've got to do sequencing, mRNA profiling, genotyping, microRNA profiling, I mentioned earlier, epigenomics, proteomics. So these are all the different aspects that one needs to look into a patient, not just sequencing the genome, but also these other aspects that need to be profiled. And then we can have that data pooled into one, integrated database. Let's say if this is a hospital or a university health system, then all that needs to be integrated into one pool. And so we can standardize the data across the board, and manage the resource, manage the storage, annotate the data accordingly for following certain standards that have been defined, and then analysis can be made easy and maybe automated and then visualized, maybe on the fly, right? So if we have something like that set up, then we can really maximize from this. The problem is a lot of times this is all scattered, locked into different departments, no sharing between departments. I would prefer this is a national effort, top-down government, government mandate that we got to do this. That will be the best. Then it gets implemented easily. But if it's from bottom up, we need both, of course. But from bottom up, usually it's much more challenging. And half of the time, it's the data is there, but just not put together properly. So So what's the solution? How can we do it the best way possible? So this slide kind of gives that example. So you have data from various sources coming together, integrated, and the bioinformaticians are sitting at this data integration box here in the green, and they're going through all that data and trying to annotate them, present them, and facilitate analysis of the data. And of course, that's not, molecular profiling is not the only data that comes to this integral resource. We also have the clinical and pathological data. We also have the experimental data, for example, treatment and responses in xeno patients at the cellular level, and of drug screening at the cellular level. So when you have data from, you have experimental data, you have the molecular profiling data, you have the clinical and pathological data, all that comes together. Imagine the richness of your data, and you can just do so much data mining. So data warehousing, which I covered earlier, if we follow those principles, then we can nicely integrate all this, and then data mining can be done very effectively. So we can ask all kinds of complex questions and get those results very quickly without having on an ad hoc basis to repeat certain processes or certain steps. So we might, I think it makes a lot of sense to invest for something like this. So it might take time. It might take many years just to get this done. But once it's done, mind you, I think you are slated to be able to really reap maximum benefit from such kind of a development. So it really makes sense to invest and to think about some sort of a setup like this. I have seen something like this implemented in the West, especially in Europe and in the US. I think in Asia also there are countries that are moving in this direction. China, Japan, I think Singapore also with their precision medicine initiative, they're spending billions. I've heard from conversations that trying to come up with something like this. So I'm not sure about Indonesia, but I hope maybe Riau could explore something smaller scale for this, or the government could be maybe it's thinking along this line. But this is something really worth considering. And if imagine this is not just your university or your hospital, but actually all the hospitals or every health institute in the country is part of this initiative, then there's really a lot that we can gain out of this. And at the end of the day, who's going to benefit? The patients are going to benefit out of this. So yeah, it makes it really facilitates precision medicine and candidate prioritization, like identifying drugs or vaccine targets or diagnostic markers to be deployed, makes a whole lot easier. I mean, this might look like a schema, a plan, but actually this is already being implemented. This is just one example of a bioinformatics pipeline where various sources and tools are put together to extract value, and with applications to diagnostics, cancer treatments, analyzing predisposition or prognostics, pharmacogenomics, infertility and IVF. So these are already some of the applications that are already seeing the benefit from this. Of course, there's more to be done, but it's nice to see that this is already happening. This is just a simple, example case of how we don't even have to have a sophisticated setup like this. Even a simple setup, aiming for the low-hanging fruits can already give us very promising results. So this is a story of a boy, Nick Walker. He's a 6-year, I don't think he's 6-year-old anymore. So who developed inflammatory bowel disease, and he had wounds in the intestine. He was leaky, a lot of visits to hospital at the age of 4, and cause of disease was unknown. He was written off, but it's a genome, specifically exome sequencing. So this is very cheap these days, and I mentioned like 5 minutes, and very price-wise is one of the lowest out there. Not even compared to whole genome sequencing, this is much cheaper than whole genome sequencing. So very low cost, and very quickly you'll be able to get the data, and then you can start analyzing the data. And when they did analyze it, they found a specific mutation in this gene, and through bone marrow transfusion, they were able to treat the boy. And I'm not sure how is he doing now, but it seems, based on what I've heard, that he kind of recovered, or at least was able to manage that problem, that issue. So here's a promising outcome with just addressing the low-hanging fruits of such a challenge. And imagine what we can do if we are able to implement such a setup. So the promises are many. You can diagnose your disease, pinpoint genetic factors that cause your disease, predict how severe your disease might be, choose the best medicine, the best dosage, and discover factors that might increase your disease risk, find genetic factors that could be passed down to your children, screen newborns for certain treatable conditions, and maybe many other endless possibilities. And of course, all this, if this can happen, I think if we maximize the value, I think this is definitely contributing to the green approaches. And I want to very quickly touch up on something much larger beyond precision medicine that's actually happening out there, which is digital health. This is not just taking aspect of genetic differences and precisely identifying what exactly is the problem for a particular disease. This is taking it a whole different higher level, and this can be defined as the use of information and communication technologies to monitor and improve the well-being and health of patients, and to empower patients in the management of their health and that of their families. And of course, precision medicine is a key part of that, but this is the larger landscape, and I think it's important as part of this today's conversation that we also recognize this initiative. So I will quickly just touch on this. just a lot going on. Of course, big data is there. You have your EHR, AMR databases out there, collecting patient data. Interoperability is important. Personal genomics is still there, which I've covered. And there's a lot more that's happening, and that's really digitizing health and trying to bring the maximum value out of this health industry. So there's innovation that's actually centered on the big data. So a lot of this is really centered on the fact that we have a lot of data, and so we have all kinds of innovations from research, telehealth, telecare are some of these, logistics apps, medical apps, health apps, and algorithms and AI are also some of the connectors of all these, and sensors are coming into the picture, so we can really reap the maximum from integration of all these technologies and innovations. So it's an exciting time in healthcare at the higher level, not just precision medicine. So the new world versus out world, payment would be outcome based. You don't just pay as a fee for service, but really at the value that you're getting, and the incentive is the value. The focus is population health. The provider's role is not just one-off, but really a continuum, maybe from birth, maybe to cradle to the deathbed or to the grave, all the way. So information, not just looking back, but also predicting, what's going to come up. So this is the exciting time that we are excited about. The growth opportunities include improved treatment, efficiencies, and saving, which is a big aspect of green approaches. Personalized medicine, I've touched upon that. Population health, well-being of the population, and something like this would be critical at a times like this during COVID pandemic. And of course, healthcare research will bring about newer innovations, society engagement, improve well-being, improve outcomes. So this is just some of the opportunities. Of course, there are obstacles. Digital health also has its own obstacles. Data silos, I've addressed on this. Resistance to change. Some people are not just willing to move in this direction. Intellectual silos. We have the talent, but people are just, people just prefer to just keep it to their own groups or their own labs or their own institutions. Budgetary silos, proprietary systems, evidence and validation limitations, monetization of these health applications, and locality specific issues, problems specific to a certain locality. There are a lot of catalysts to overcome these problems. I'm not going to go into the details, but people are working on how we can overcome these obstacles. And in conclusion, I hope I just made it. So I hope I have convinced you that bioinformatics underpins biomedical research, or probably you were already convinced. I hope I gave you some examples of how it does that. And precision medicine is a key goal of the healthcare industry, is one of the key ones. But there are other goals of the healthcare industry, and digital health, the bigger initiative, the larger picture is trying to glue all that together. But it has its own challenges. So digital health adoption is going to be a key challenge in moving, countries or cities in that direction. And to overcome these challenges, we have we need to bring all the stakeholders together and be part of this process. So from industry, academia industry partnership is key, but this really needs to be done with care, because there are some sensitivities that go against each other. Industry might want something, academia may not. So we got to find some common grounds where we can agree upon. Of course, in order to drive this, we need some in-house champions. Riau might be able to drive some of these and bring forth some exemplars that could be used to attract others to go in that same direction, and maybe get the support from the government funding agencies to be able to do more of those things. So we need to focus on some key exemplars as champions for digital health at the university and beyond. So in general, we can look forward to better days for our patients, and of course all this I'm sure will contribute to the sustainable development goals that I mentioned previously, the number 3, which is on health, number 12, which is on production and capacity, but also directly and indirectly into the other SDG goals. So with that, the last 2 slides, I just want to say that data is everywhere. Really, this is a time of data deluge, and with a lot of opportunities. We have low hanging fruits and higher hanging fruits. And of course, without much investment, we can already reap the fruits from the low hanging fruits. But we got to be prepared. We got to look out for these opportunities. We got to build in the infrastructure, train the people to really capture all that opportunity and see the outcome of that effectively. And so critical need for bioinformatics expertise, we need to train our people to be able to do these kind of things. And there's a big demand for bioinformaticians, and we don't have enough supply. So I hope Riau University could actually look into training people in this area. So I just want to end with this quote from one of the papers that I showed earlier by David C. Pension. The future is bright only if we address our current obligations seriously and systematically. And I hope bioinformatics can be that that ladder that helps do that systematically in the area of medicine and life science research in general. Thank you very much.
Okay, thank you very much, Dr. Muhammad Asif Khan, for your concise presentation. So we go next for the Q&A. Is there any questions from the participants? Okay, I think that's a lot. There's so many. So the first one is from Alma Ashila from University of Lambung Mangkurat. Does that mean that green approaches and sustainability efforts are in favor of open access in knowledge? And what do you think of the future of research publication?
I am in favor of open access. So I think I personally think knowledge should be made available, made accessible. There's a lot of debate I know that's going on in this. But the good news is, I think in general, we are moving in that direction. Knowledge in general is becoming more open, more accessible to people. Nowadays I can read papers much more easily than back then in the olden days. So I think it does go hand in hand and is in favor of open access. That's my personal opinion. But yes, there are challenges. We don't have all papers available right away, and the publishing agencies have their reasons why they want to do that. But I think there are a lot of advocates for open access, some very serious on this, and some funding agencies have actually come to the fore and listened to these complaints, and they have made it much easier that at least papers that they are funding should be made available publicly. So I think I agree. Yes. So in a nutshell, I think they go hand in hand and support open access in knowledge. And I think the future of research publication will be more open access. It's going to take maybe more time, but I think we can already see the results of the various struggles of key people out there who are pushing for this agenda very strongly, and funding agencies have heard of them. So I believe this is just going to get better and better. So I hope that addressed your concern.
Okay, thank you, Doctor. Okay, for the next question from Maria Lucia Dwi Gamapurba from Universitas of Riau. Do you think big data should be publicly available?
Okay, so big data is, of course, a general term. I didn't do into the definition of big data, but it's a moving definition. So like what might be big data today may not be big data tomorrow. Big data today, if your standard tools cannot handle, that's big data. If your Excel cannot handle, that's big data. But tomorrow, whatever we couldn't handle today, we can now handle it, then that becomes the standard tool. Let's say R and all these become standard tool and we can handle big data, but data grows much bigger and then we cannot handle those. So then that becomes big data, and what we call big data today may not be big data tomorrow. So it's a moving definition. In general, it's basically anything that our standard tools cannot handle, and standard tools change from time to time. Okay, so that's the definition of big data. So do you think big data should be publicly available? I think a lot of big data is already publicly available. You just need to go and look out for them. If you go to NCBI, there's like, there are probably petabytes of data already available there for free We don't have to search for them. I mean, in fact, they make it so easy for you to even search and even download. You can download from the web, you can download from FTP. They even provide APIs for you to download. So there's a lot of data publicly available. Of course, some data is not publicly available. Those are private data, and they have reasons why they are private. But I'm also hearing that slowly these private initiatives of genomic, for example, genomic sequencing of patients, they are also working in towards making that publicly available slowly as they have a good handle of the data. Of course, one of the reasons why they don't is because of personal identifiers. So we are trying to find ways when it's patient data, how we can strip off the personal identifier so that the data can be used for research purposes. So there are a lot of efforts towards that, and actually data is becoming more and more publicly available, and there's already a lot out there already. That's big data. But in terms of patient data, we got to be a bit more careful because this is sensitive data, and we got to handle this with care. There are actually some resources that provide this sensitive data, but you got to register, you got to become a member, you got to follow the rules, then you can get access to this patient data. So there's a lot to explore, and let me know if you need if you're looking for a certain particular data, we could take that conversation on the side and explore that further.
Okay, thank you. I do agree with you. I mean, not all the data can be publicly. It depends on how confidential, I mean, the data are. Okay, for the next question is from Muhammad Abdul Karim from Universitas Riau. What kind of bioinformatics research occur today besides genomic research?
Okay, of course, genomic research kind of is the poster boy. It takes away the shine, takes the shine. So the spotlight is on that. But of course, you have proteomics research. I mean, there are various ways of classifying. You have metabolomics, you have lipidomics, so proteomics, regulomics, and immunomics, you name it, there are all kinds of research.
With nomics at the end, right?
Nomics at the end. I mean, I like to call my research interest vaccine informatics, viral informatics, venom informatics, you put informatics there. So all kinds of research is happening. It's really a fertile ground, a lot of novel approaches, novel techniques, all kinds of interesting developments are happening. Genomics is not the only front. And of course, brain research is a big deal. Recently, I attended a talk, and now we not now, but for a long time we have the term neuroinformatics. So neuroinformatics, bioinformaticians who do brain studies, or the spinal cord, neurons, neurology, neuroscience studies. So yeah, various areas. Brain is a very exciting area. There's a lot of development. Not many people are working on that. So that's something also worth exploring. But yeah, as I said, metabolomics, lipidomics, yeah, all kinds of areas. I think that kind of gives you the gist of it.
Okay, thank you. Okay, for the next question is from Sony Anata from University of Riau. I think he has 4 questions. Yeah, I think it's many. First is at the beginner, how I can start the genomic or other omics research. And the second one is and where I can find the gene database for my genomic research. The third one, what the problems that will occur in bioinformatic research. And the last one, what kind of software and database that commonly used for the bioinformatic research. I think this is especially for the free one.
Right. So a lot of questions. So thank you so much. These are good questions. Can you go back to the question?
Yes. Can you? Yes.
You moved away from that slide.
Can you go back to the next slide?
Yes. Okay, perfect. As the beginner, how can I start the genomics or other omics research? Of course, as a beginner, I think you need to first understand what bioinformatics is all about. There's a lot of material you can find online or YouTube videos, or if you need more help, I can direct you to some of these. I think first is to maybe get a good grasp of what bioinformatics entails. So when you have that big picture, maybe a lecture on bioinformatics, 1-2 hours, gives you that big picture, then you can better ask that question, whether you want to go to genomics or some other. So you also need to tie it with your own personal research interests. Where exactly do you want to go? But doesn't matter whichever omics you want to go into, you've got to understand what bioinformatics is all about. So bioinformatics, as I gave you the definition, there's data warehousing, there's data mining. So data warehousing, if you can learn a bit about data warehousing, if you can also learn or do some research work where it teaches you about data mining, then I think you will have much more clarity on how you can do this research in a bigger way. So, if you have projects that people are offering where you get some experience in data warehousing, playing around with data, putting data together, cleaning the data, or just do one, actually a simplest one would be to just do a bioinformatics research project. Usually it would have a data warehousing aspect and it would have the data mining aspect. So for now, don't be choosy. If you get an opportunity, just find an opportunity to do a research project. It's much better than listening to lectures and attending talks. It's just to immerse yourself in a research project, 6 months, 3 months, doesn't matter. If you can find such a project, then, and that has both components, then I think you're set to benefit so much more than, listening hours and hours of lectures. Immerse yourself. So I would suggest do a research project. That's the best way. Don't be choosy, choose whatever. And just get started, and then later you can decide which area you want to go to. But of course, do it with somebody who understands bioinformatics, can teach you the data warehousing aspects and also the data mining aspect. So I hope I've given you something. I'm sure there are a lot of people in Indonesia whom you might be able to find who can help you with this. The other good thing about bioinformatics, you don't have to be physically at that place. I know COVID times we can't travel. You can do this research virtually. You can have conference call via Zoom with your supervisor, and you do the work, and then you can show the work. So traveling is, it's not a requirement. You can do this remotely, and that's one of the best aspects. I love to joke that you can do bioinformatics as long as you have a digital device or smart device. Even a phone is good enough for you to be able to do bioinformatics research. And you can do it from anywhere as long as you have internet and a smart device. You are all set. Of course, a laptop would be much better. So where can I find the gene databases for my genomics research? There are many places. You can Google, of course, but there are something I like to call resources, or this, you can call them catalogs of databases. So if you look at the NAR database server issue, NAR web server issue. So NAR is a journal, Nucleic Acids Research Journal, high impact factor. Every year they publish a database issue, they publish a web server issue. So, and they actually have been compiling all this into one website. So you can Google for this NAR database issue, NAR web server issue, and then find the link. If you're struggling, you can reach out to me, my email I provided earlier, and I can direct you there, but you should be able to find it by yourself. This is just one. There are others. There's a journal called Databases, I think, or Database, and you can find many from there. I think a Google search should be able to get you. Look for like bioinformatics database catalog, bioinformatics database resource, or bioinformatics tools catalog. You should be able to get a good start from there. Number 3, C, what problems will occur in bioinformatics research? Well, like I mentioned earlier, we spend a lot of time just cleaning the data. So be prepared for a honeymoon with the data. Okay, it might be 3 months, might be 6 months, depending on how clean is the data. I had a student who spent 1 whole year honeymooning with the data before even doing any analysis. So be prepared for that, and don't rush it, because it's important that you really have a good handle on your data before you do any analysis. Otherwise, garbage in, garbage out. You don't want that. So have patience and make sure that you have a good handle on your data, and make sure you know the processes of cleaning the data, what to think about when cleaning the data. This is where you need a supervisor who has a good handle on the data, so that he or she can guide you in terms of what's the best way to clean, and then before you do your data mining. What kind of software, freeware database commonly used for? Well, it depends on your research question, but NCBI is a big resource where a lot of people go because it provides a lot of databases. NCBI is not a database, it's just an organization. They have a website and they provide a lot of these databases. A lot of them are for free. Some of them you may have to register, but that's not a problem. Of course, the European side also, European Bioinformatics Institute also provides a lot of these resources. Most of them are for free, and there are many others. I think DDBJ from Asia, Japan also has a lot, and there are others who provide this. So go to the the catalogs I mentioned, NAR database catalog, NAR web server catalog, and that's a good start for you to understand what kind of software are available. Many of them are free. There are of course commercial ones, but you should be able to do many of the things you want to do from this free software, and there are many databases out there for you to use. So yeah, the problem is not the availability of data or the databases or the tools. The issue is, are you ready to dive in and explore the world of bioinformatics and try to solve some real challenging questions out there. I hope I answered all the four.
Okay, thank you very much. I think it's with the time constraint. I think we've stopped our question Q&A now. Thank you very much for Dr. Muhammad Teşekkür ederim. I think if I'm not mistaken,
yes, that's
Turkish.
correct.
I hope you enjoyed the webinar for today, and I hope you can come to visit University of Riau in the further times.
Inshallah, yeah.
Okay. And this is the end of morning session of this webinar. And then on behalf of the committee, I would like to thank you all the honorary guests, speakers, and participants. And thank you again.
Thank you very much.
Yes, I hope. I think this is the early morning in.
Yes,
Istanbul,
the sun has not risen yet.
Yes. So I think you're gonna be sleeping and wake up very late today. Yeah. Okay. Thank you.
Okay. Thank you.
I will give it back to the host, and thank you again, and good afternoon.
Okay, thank you very much for our moderator, Dr. Rudy Hendra, for giving us to lead the lecture. Okay, the next event is the introduction video from Chemistry Department and Riau University. So let us enjoy the video.
Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Faculty of Math and Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Second is bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilman Yuda Teruna, Dr. Rudi Hendra, Dr. Dewana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Rino Novianti, and Yuharme. Drug Design and Organic Synthesis, led by Professor Adel Zamri, Professor Jasril, and Dr. Yum Erianti. Fourthly, is Petroleum and Biofuel Chemistry, led by Dr. Sofia Anita and M. Rizal Mahidin Tambusai. Fifthly, is Adsorbent Coagulant and Biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halsayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itna Wita, and Muklis. Our students have received numerous awards, such as funded research by the Ministry of Education, Professor runner-up of UDC 2019, second runner-up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us.
Okay, that's our video. And that's our last schedule of this session. But before all participants left this Zoom meeting room, we will take some documentation. So please, to all participants, to turn on their camera. And the committee will take the capture of this Zoom. Okay, I will count down from 3 until 1, and then the committee will capture. Okay, make sure all participants has turned on their camera. Okay. Okay, we're waiting for the other participants to turn on their camera. Okay, 3, 2, 1, cheese. Okay, maybe another once. We'll take a picture once more. Okay, I will count down from 3, 2, 1, cheese. Okay. Thank you very much for all the participants that has been gathered here. That's all about our first session of this international webinar. And don't forget to fill the questionnaire form and the chat box. And as a reminder, don't forget to, we are still have the second session at 1 p.m. UTC +7. So make sure you come back again at the second session if you have been registered at the second session. Okay, thank you very much for your attention. My name is Sonia Nanta. We will be right back at the second session of the international webinar.
Hi, I'm Teresa from Spain. I'm currently living in the Netherlands. And I met Ria Rhythm almost 2 years ago, thanks to a project called Vínculos, organized by the Orchestra Aucas from Spain, and it's nice to play with you guys again. Thank you.
Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Faculty of Math and Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high-quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Is bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilwan Yuda Teruna, Dr. Rudi Hendra, Dr. Yohana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Riri Novianti, and Yuharmet. Thirdly is drug design and organic synthesis, led by Professor Adil Zamri, Professor Jasril, and Dr. Yum Erianti. Petroleum and Biofuel Chemistry, led by Dr. Sofia Anita, and M. Rizal Mahidin Tambusai. Is Adsorbent Coagulant and Biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halzayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itnawita, and Muklis. Our students has received numerous awards such as funded research by Ministry of Education, Novice runner-up of NUDC 2019, second runner-up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us. Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Second is bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilwan Yuda Teruna, Dr. Rudi Hendra, Dr. Yohana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Riri Novianti, and Yuharmet. Thirdly is drug design and organic synthesis, led by Professor Adil Zamri, Professor Jasril, and Dr. Yum Erianti. Fourthly is petroleum and biofuel chemistry, led by Dr. Sofia Anita and M. Rizal Mahidin Tambusai. Adsorbent coagulant and biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halzayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itnawita, and Muklis. Our students has received numerous awards such as funded research by Ministry of Education, Novice runner-up of NUDC 2019, second runner-up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us. Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Faculty of Math and Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high-quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Is bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilwan Yuda Teruna, Dr. Rudi Hendra, Dr. Yohana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Riri Novianti, and Yuharme. Drug Design and Organic Synthesis, led by Professor Adel Zamri, Professor Jasril, and Dr. Yum Erianti. Fourthly is Petroleum and Biofuel Chemistry, led by Dr. Sofia Anita and M. Rizal Mahidin Tambusai. Adsorbent Coagulant and Biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halsayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itnawita, and Muklis. Our students has received numerous awards such as Funded Research by Ministry of Education, Novice Runner-Up of NUDC 2019, Second Runner-Up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us. Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilwan Yuda Teruna, Dr. Rudi Hendra, Dr. Yohana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Riri Novianti, and Yuharme. Thirdly is drug design and organic synthesis, led by Professor Adil Zamri, Professor Jasril, and Dr. Yum Erianti. Fourthly is petroleum and biofuel chemistry, led by Dr. Sofia Anita and M. Rizal Mahidin Tambusai. Adsorbent, coagulant, and biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halzayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itnawita, and Muklis. Our students has received numerous awards such as funded research by Ministry of Education, Novice runner-up of NUDC 2019, second runner-up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us. Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Faculty of Math and Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high-quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Is bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilwan Yuda Teruna, Dr. Rudi Hendra, Dr. Yohana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Riri Novianti, and Yuharmet. Thirdly, is drug design and organic synthesis, led by Professor Adil Zamri, Professor Jasril, and Dr. Yum Erianti. Fourthly, is petroleum and biofuel chemistry, led by Dr. Sofia Anita and M. Rizal Mahidin Tambusai. Fifthly, is adsorbent coagulant and biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halsayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itnawita, and Muklis. Our students has received numerous awards such as Funded Research by Ministry of Education, Novice Runner-Up of NUDC 2019, Second Runner-Up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us.
participants, please stand by. For all participants, because we will enter into the second session. Thank you. Please stand by for all participants because we will enter into the second session. Thank you. Please stand by for all participants. We will enter into the second session. Thank you. Good afternoon, ladies and gentlemen. The Honorable to the Speaker Dr. Raihana Begum from Nanyang Technological University, Singapore, and Professor Dr. Harris from University of Groningen, Netherlands. The Honorable Dean of Faculty Mathematics and Natural Sciences, Dr. Shamsuduha. The Honorable Head of Chemistry Department, Dr. Muhdarina. And welcome to the second session in this international webinar, Green Approaches in Medicine, Chemicals, and Energy, in conjunction with the 58th anniversary of Chemistry Department, Faculty of Mathematics and Natural Sciences, and Riau University. My name is Muhammad Abdul Karim, chemistry student of Riau University, as your second host for today. For all participants, before we move to the main event, please fill the attendance form, and I remind you, this attendance is only for participants who participate in this second session, so that we can mark your attendance for e-certificate. Again, I remind you, this attendance is not for the participant who participate both of this webinar session, and at the end of this event, the committee will send the questionnaire that will be filled by participants, which is the link will be sent on chat box. And for participants who are in the YouTube, you could be write the link and pass to your browser, so we can mark you as participant in Zoom. And let me show you the scheduled event for the second session, which is you can see on your screen. Okay, thanks. We move to, I would like to show you the video of role in this webinar. Please enjoy the video. Okay, I think it's enough. Let's move to our main event. Start by moderator, a lecturer from Chemistry Department, Riau University, and the speech will be delivered by our speaker, Dr. Raihana Begum and Professor Dr. Harris, with the topic is not less important than others. Without any further ado, let's welcome to our moderator, Dr. Leo Saputra. The stage is yours.
Okay, thank you very much. Good afternoon. Thank you for joining the second session of this webinar. Distinguished speakers, ladies and gentlemen, it's a great honor to be a moderator of this session. I am Leo Saputra, a lecturer at the Chemistry Department. Our first speaker of this session is Dr. Raihana Begum, a young and excellent scientist from India. Dr. Raihana is a research fellow at NTU SCARES, Nanyang Technological University, Singapore. She got a PhD in chemistry from IIT Guwahati, India, on engineering optical properties of quantum dots by chemical reactions. In 2015 to 2017, she was a postdoctoral fellow at KAUST Solar Center, Saudi Arabia, on the fabrication of perovskite semiconducting nanocrystals. She already published 16 papers in highly reputable journals, including JACS, the Journal of American Chemical Society, RAC Chemical Communication, and Nature Communication, with a total citation score of 903. She also got Young Scientist Award from Materials Research Society of India. Today, she will deliver a talk entitled Colloidal Semiconducting Quantum Dots from Synthesis to Applications. For participants, you can type your questions on Zoom's chat box or comment on YouTube during the presentation. And you can also directly ask the speaker by clicking the right hand button on Zoom during the Q&A session. And don't forget to mention your name and affiliation. And Dr. Raihana, how are you today?
Thank you. I'm good.
Okay. Okay. Dr. Raihana, you have about 45 minutes for presentation and 15 minutes for Q&A session. Okay. Let's give a warm welcome to Dr. Raihana. Okay, Dr. Raihana, time is yours.
Thank you. Thank you so much, Dr. Leo. Is my voice audible?
Yes, clear.
Okay. Okay. I share the screen. Share screen is done. It's coming, right? The screen share is coming, right?
Yes, yes.
Okay.
Okay.
Thank you. Thank you, Department of Chemistry, Riau University, for giving me this opportunity to present my work before you. Actually, it's the first time for me to talk before young students. That is why thank you so much for giving me this opportunity. So today I'll be talking on this topic, colloidal semiconductor quantum dots from synthesis to application. Before that, I'll be talking a little bit about nanomaterials and quantum dots, a basic introduction. And after that, I will talk about my postdoctoral research. Where I have 1 year postdoc experience from KAUST Saudi Arabia and a few years in NTU Singapore. And my work, postdoctoral research work, is mainly based on lead halide perovskite quantum dots. And I will also show some of the LED applications. And my PhD work in IIT Guwahati, India, where I have worked on chalcogenide quantum dots and bioimaging of cancer cells. Okay, so I was looking for some examples to give some relevant examples for relative size of the particles. So I found this one that was published in a Visual Capitalist just few weeks back. So to give an example of size of nanoparticles and their sizes, I think during this time of pandemic, the size of the coronavirus. So I was curious, like, okay, what's some relevant example can be coronavirus. So what is the size of this virus? So I found, so for this picture, it says that the size of the coronavirus is about 100 to 500 nanometer, and the respiratory droplets containing the virus could be about 5 to 10 micrometer. And we know the size of red blood cell is about 7 to 8 micrometer, and the diameter of human hair is 50 to 180 micrometer. So, this is one of the rare picture that was published by New England Journal of Medicine that shows the transmission electron microscope or microscopic picture of coronavirus. That is, from the scale you can see that it is about 100 nanometer. And there is another article that says that the spike proteins on the surface of the coronavirus is about 10 nanometer. And then I found another very relevant information like the iPhone 12 that was released just a few days back. The iPhone 12, this is not a promotion, by the way. So I just wanted to say like how a 2 nanometer difference in the size of the chip can cause a large difference. So like the iPhone 12 has the A14 Bionic chip that is 5 nanometer in size, and the iPhone 11 is 7 nanometer in size, and just a 2 nanometer difference can cause 83% of more efficiency. It's more faster. It has like the 5 nanometer chip has like 11 trillions of it can perform like 11 trillions of operations per second, while the 7 nanometer can perform 6 trillion operations per second. So is it just about the size? The 2 nanometer difference causes so much of size. So if you see like why such a difference in the performance when there is just a change in the size by 2 nanometer. So as we know, nanoparticles are the particles that are smaller than 100 nanometer. They shows novel physical and chemical properties that is different from bulk material. Bulk material, that is anything that we can see with our naked eyes. So what happens when the size decreases? So suppose this is a box and you cut it into 2 pieces. So when you cut it into 2 pieces, you generate 2 new surfaces. So the surface area increases. Suppose, so here on the left, we have 3 boxes. One is 1 centimeter, 2 centimeter, and 3 centimeter. Now, if you calculate the surface area and volume and take the ratio, then we find that the surface to volume ratio is very high for the 1 centimeter, is like 6 to 1, while for the bigger box, it is 2 to 1. So this is just in a 1 centimeter scale. However, if you go to the nanometer scale, the surface to volume ratio is very high. So this is another a very good picture that shows gold nanoparticle. Like we know, bulk gold is yellow in color, but when we make nanoparticles, they shows like purple color, red color, and depending on the composition, like this is the first one on the left is AU gold nanoparticle. This is silver nanoparticle, and if you have an alloy of gold and silver, it looks like light purple color. So depending on the composition as well as their structure, they shows different color. Or is it just about the color or the size? No. So when because of this their chemical properties changes, that is because of the large surface to volume ratio, and the electronic properties changes, like the quantum confinement, the band gap energy changes, and the density of states, electronic tunneling, etc. Also, if we suppose dope some magnetic ions like manganese in these nanoparticles, it shows magnetic properties as well. So as well as it shows different mechanical and optical and thermal properties when you change the bulk particles to a nanoparticle size. Is my voice okay?
Okay.
Yeah,
no problem.
okay. So this is a simple application of the synthesis of nanoparticles. So there are two approaches broadly, that is top-down approach and the bottom-up approach. So in bottom-up approach, so you add the precursor materials and then it undergoes chemical dissociation, and from those precursor material you form the nanoparticles. However, in the top-down process, suppose you have a bulk material, you cut it down into small like powder-like structure, and from that you make the nanoparticles. So some top-down methods, examples are like lithographic methods, laser pyrolysis, and even like mechanical grinding is a example of top-down method. So, like in microelectronics study, in microelectronics industry, lithography is mostly employed to make those electronic devices, the electronics chips and the transistors. Yeah, and some example of bottom-up method is like chemical synthesis by wet chemical methods, self-assembly, and electrochemical reduction methods. Those are example of nanomaterial examples of bottom-up method. So these are some examples of different heterostructures formed by bottom-up synthesis. Some examples like nanorods, nanowires, nanobells, and nano templates. So from the scale bar, you can see like a nanowire, for example, is several hundred meters long and the diameter is very thin. So similarly, like nanoplatelets are like few hundred meters in length and the width. However, the quantum dots are just like a dot. And if you see the first picture on the picture on the high resolution picture on the left, it is just 5 nanometer in size. So what are the quantum dots? So my research work is mostly focused on quantum dots since my PhD. And so I would like to give a brief introduction of the quantum dots. So quantum dots are zero-dimensional semiconducting nanocrystals, and their size is 1 to 10 nanometer. And they are characterized as the binary, ternary, and quaternary. So for the binary quantum dots, like you can, some examples are zinc sulfide, cadmium sulfide. So this is mainly from the group 2B and group 6A. So you pick any metals like from zinc and then sulfur, or cadmium, then selenide, or mercury and tellurium. So this kind of combinations are used for making the binary quantum dots. And for ternary, you choose 1, 3, 1, 3, 6. So 1 is the copper, or even the copper, silver, gold. Copper, silver is mostly used, and 3 is in this one group the aluminum or indium, and the 6th group is mainly the sulfur and selenium, those kind of, and quaternary is also like that. And besides this chalcozenides or nictozenides quantum dots, there are also the carbon dots, silicon quantum dots, and graphene quantum dots, and very recently in 2014, the lead halide perovskite quantum dots were introduced. The quantum dots were first discovered by Louis Bruce in 1980s, early 1980s, and it was believed that the quantum dot, the name was given by Mercury. And the first paper appeared in 1981, and then in 1988, this paper says that zero-dimensional semiconductor nanostructures. You already, the students in the MSc master degree, they already know the band theory in solids. So you know, like in insulator, you have a large forbidden gap. Therefore, the electrons in the valence band cannot jump to the conduction band. However, in the metals or in the conductors, there is practically no forbidden gap, and therefore it is a good conductor of electricity. However, in the semiconductor, the bulk semiconductor, for example, in silicon, germanium, or gallium arsenide, these are some of the known widely used semiconductor. where there is a reasonable forbidden gap, and when it absorbs energy, it leaves behind a hole in the valence band, and the electron leaves the valence band and goes to the conduction band, and leaving behind a hole. Now, this is a very common term like excitant. So this electron in the conduction band and the hole it leaves behind in the valence band, that electron hole pair is called excitant. And there is another term that is excitant Bohr radius. So excitant Bohr radius is the distance between the electron in the conduction band and the hole in the valence band. This Bohr excitant radius is like a fingerprint. Like for every semiconductor material has a different Bohr excitant radius. So because this is a bulk semiconductor, like silicon, a piece of silicon or a silicon wafer or something like that. So because it has a large volume, therefore this excitant, it is the electron and the hole pair, it has a large volume to occupy, and therefore it gives rise to some continuous energy band. Now, this is another depiction of density of states. So by definition, it is the number of states per unit volume, per unit energy. So in this is a graphic schematic representation, like in bulk material, the motion is not confined. However, in a two-dimensional, suppose a quantum well, the motion is confined in two-dimensional. Similarly, in a wire that is one-dimensional, the motion is confined in one-dimensional. However, what happens in a quantum dot? The motion is confined in all the dimensions, all three dimensions. And as a result, if you see the density of three-dimensional is represented by the solid line, so it is like a band. And for 2D, it is like a staircase. And for one-dimensional, it is this one. However, for the zero-dimensional, it is the black lines. It's just like some perfect lines. Okay, now quantum confinement. So these are some basics of quantum dots. So before I go to the main part, these are just basic introduction. So what is quantum confinement? Now, if we say quantum confinement, so it is the confinement of the, is there any question? Oh. Chat. Okay.
Please continue your presentation.
Okay. Okay, so quantum confinement is the confinement of the charge carrier, like electron and the hole. The electron is negatively charged, the hole is positively charged. Now the confinement of the charged particle in a dimensional that is smaller or equal to the de Broglie wavelength. So, or in other words, you can also say that this is the reduction, this confinement is a reduction of in the degree of freedom of the charge carrier particle. So yeah, so in a quantum dot, because it has only few atoms, so the excitance or the electron hole pair is confined in a very smaller space that is equal to the excitant Bohr radius is equal in a space that is equal to the excitant Bohr radius. Yeah. So and because of this, like in bulk, you know, like it is a band. However, in quantum dots, also known as nanocrystals, so the band is split into discrete or like quantized energy levels, like proper lines. So, and this is similar to an atom. Therefore, these quantum dots or nanocrystals are also nicknamed as artificial atoms. So, and there is another thing that when the size of the quantum dot is, you change the size and the band gap energy also changes. Suppose if it is a smaller, suppose one is 5 nanometer in size and another is 2 nanometer in size, then the band gap energy will also change depending on the size of the nanoparticle. So what are the methods of methods of tuning the optical properties or other properties. So so this is image of quantum dots ranging from 1 to 10 for cadmium selenide quantum dots. So as you can see, from 2 nanometer to 10 nanometer, the color, the emission color, this is under UV light. So these nanoparticles, the color changes from blue to red. And similarly, if you change the composition of the nanoparticle, it also changes the color. Similarly, suppose you dope some transition metals like manganese or copper in this in this quantum dots. That is, suppose here the example is zinc sulfide and zinc selenide, which is the host, and manganese is the dopant. So if you have, like you can see manganese dope in zinc sulfide and selenide, the color is little bit different. All here also on the right side, even though the host is the same, like zinc selenide, however, is the dopant is different. Like here it is manganese, here it is manganese, and here it is copper. So depending on the dopant, the color, the emission color is different. This is also under UV light. So similarly, the host is the zinc sulfide and the dopant is manganese or copper. And depending on that, the emission color is different. Now, why does it the color change and what happens upon doping? So this is the energy level of the host, the valence band and the conduction band. And normally we have some trap states in between. So normally, like from here, the electron will jump to the conduction band, and normally it will recombine, and then it comes to the lower energy, to the valence band, to the ground state. So when you dope with some metals like copper or manganese, it introduces some energy levels within this host energy levels, like within the conduction and the valence band. It will introduce some new energy levels, like for manganese, it is 41 to 6A1, this energy level. So the hole is here, and electron it leaves behind the hole, and electron goes to the conduction band. Now this will go through. So otherwise, like it follows this path. Now when you have a manganese, it will follow this path. And as a result of that, suppose it is only zinc sulfide, then it will give an emission peak at around 400 nanometer or 410, like that. However, if you have a manganese, it gives an emission around 600 nanometer, around like 590 to 600. And depending on the concentration, suppose if you dope with different percentage of manganese, then the peak will be different. Like there is an optimum concentration where it gives the maximum quantum, or like it is more intense or more emissive. So here in this paper, they show that 2% of manganese gives the highest emission. So this is again another example where like the dopant is copper, and you have different hosts. The host is zinc sulfide, zinc selenide, zinc cadmium sulfide, like that. And depending on that, because the host is different, so emission color is also different. Like you can tune it from like 440 to up to 670. Like if you have copper doped in indium phosphide, then the color is towards the near infrared. Similarly, nickel doped in this is also in zinc selenide or cadmium zinc sulfide. The color is different. So here also, how to check the. Okay, this is not for me, right? Okay, questions are not for me. Okay. Okay. Yeah. So here also for the same host, different dopants like copper and manganese gives different color. So, and another thing, like why it shows different color, or why how the surface area matters. So like from this figure, you can see the smaller is the size, the higher is the surface, or so is the higher is the surface energy. And also, coordination numbers. So the the coordination number also decreases, and as a result of that, there are some trap states. So these trap states are like some vacancies or like the defect state, like you have read those Schottky defects or Frankel defects, those kind of defects, or it could be like loosely bonded atoms that are like loosely on the surface of the quantum dots. So this trap state or defect state, they decrease the emission quantum yield. So because like normally from here it goes to here, the electron is on the conduction band. So if it follows the path A, that is the radiative recombination, then it should be emissive. However, if it follows the path B, non-radiative, that doesn't give you any emission, no light. So it is non-radiative. So yeah, so there are different yeah. Surface, it's fine. Okay, surface, there are also some surface phenomenon. Okay, so here, like I said, there are some, the surface energy increases and the surface area also increases. And this nanoparticle, they always have the tendency to minimize the surface energy. So that is the, it tends to minimize the surface energy. So there are two important phenomenon. One is Ostwald ripening, and another is the sintering. So sintering normally occurs at high temperature. The first one is sintering. So here the nanoparticle, they merges together, forming a bigger size. However, the Ostwald ripening is about there is a bigger particle and it has some smaller nanoparticle. So this is also some people call it like big fish. It's the small fish and grow bigger. So by definition, that the larger structure, it will eat the small structures and it will grow into a bigger size. So this surface phenomenon, we tend to reduce by passivating the surface, because we do not want a bigger size. We do not want the merging of the nanoparticles. So we want to prevent that sintering or the Ostwald ripening, and we want to prevent this minimization of the because it has the tendency to minimize the surface energy. So it wants to form a bigger particle. However, we do surface passivation to keep the size intact. And this surface passivation also helps in conjugation. Suppose you want to use these nanoparticles for bio application, then you cap it with different hydrophilic groups, because in biological cell we have lots of water. The major component is water, so we use some hydrophilic groups, functional groups like hydroxyl or amine groups, so that it goes into the biological cells. And also we modify the surface with proteins kind of thing, molecules, so that it is easy to easy to be uptake by the biological cells. And suppose instead of hydrophilic group, we have some hydrophobic groups. Suppose you want to redispose the quantum dots in non-polar solvent like hexane or toluene kind of solvent. So you will passivate the surface with non-polar groups. This is mechanism that was proposed by Lamela Danger, and it says that initially you add the precursors. They will first generate the monomer, and then it will undergo nucleation. So first you add the precursor, suppose zinc acetate or sodium sulfide, and first it will form the ions. It will get dissociated into the ions like zinc ions or acetate ions, and after that it will in the second stage it will nucleate, and then this after nucleation it will form a bigger the nanocrystal will grow. into a bigger. However, at a later stage, they tend to aggregate and also undergoes Oswald ripening. Therefore, we add the ligands or capping molecules like long chain capping molecules or some functional molecules having functional groups. This is a picture from this book where it says it shows like cadmium and selenium ions, and some organic molecules are represented by this, the blue tails, and the organic molecules act as a surfactant, and it binds to the surface of these nanoparticles. And when the crystal reaches the optimum size, then on the surface of this nanoparticle, the organic molecule, it caps it completely so that it doesn't undergo those Oswald ripening, or it doesn't undergo agglomeration, and it retains the size of the nanoparticle or the size of the quantum dot. Now, how do we characterize these quantum dots? They have normally size in the range of 1 to 10 nanometer. So how do we characterize the size? So for that, we normally use UV visible spectroscopy, FTIR, XRD, or XPS for the surface, like what is the surface chemistry, and we use transmission electron microscopy to record images of the nanoparticles. And the XRD gives different phases, like crystal phases. We get to know the crystal phases of these nanoparticles. So the quantum dots these are some examples where some in situ where bioimaging was done. So this is a mouse fibroblast cell that was labeled with cadmium selenide quantum dots. This is a mouse that was coated with 3 different quantum dots emitting green, yellow, and red. And this is also actin filaments that was stained with quantum dots. Human epithelial cells coated with quantum dots. So why do we use quantum dots and not the organic dyes? Because the quantum dots are inorganic in nature, or organic dyes. Some example of naturally occurring dyes are like turmeric or indigo, which most of our household have. Turmeric and indigo. So similarly, there are some synthetic dyes as well that we use for dyeing our clothes and for various purposes. And these synthetic dyes are also used for bioimaging and for LED devices, or like for solar cells as well. So what is the advantage of having quantum dot over the dyes? So the first is that these quantum dots, because they are inorganic in nature, like zinc sulfide or cat sulfide, lead sulfide, these are inorganic in nature. And as a result, they have very high photostability and also thermal and chemical stability. Suppose a very simple example, suppose you have some staining on your shirt by some curry that has like turmeric. So if you put it on the sunlight, the color degrades. Suppose you wash with some soap and then put and put it under sunlight, the turmeric color disappears. Similarly, suppose we give indigo color in our uniform, the white uniform shirt, and if you put it under strong sunlight, the color sometimes fades away. So because these are like organic molecules, so even under the sunlight or any other light, so they fades away. So however, the quantum dots are very stable, have very high photostability, and also they have a high extinction coefficient. Quantum yield is very high, and they have a large absorption window, and they can be because they absorb at a very broad absorption window. Therefore, they can be excited at any wavelength. And also the full width half maximum is also very narrow. And however, like for quantum dots, it is like normally 20 to 30. However, for the organic dyes, it is like 50 nanometer, very broad emission. And the organic dyes photobleach easily, and therefore they need some filters when you want to do some imaging kind of work. And some optoelectronic applications of quantum dots are like LED, solar cell, and yeah, some of this as photovoltaic model. And if we compare the lightning technologies, like we have three different bulbs, incandescent, while CFL and LED. So we see that even though the price of the LED is higher, like for incandescent bulb or CFL bulb, the price is like $1.2 or like $4. However, the LED bulb may cost higher. However, the lifespan is very high. Like this is in hours, so it is and also their efficiency is high. It consumes less power, and it consumes less, so if you calculate the cost, the annual cost, you see that this kind of LED has a lower cost when you calculate it per year. And this is from the OLED TV and QLED TV. So QLED means quantum dot LED, and OLED is organic light emitting organic LED TV. So this is an old data. I'm not sure the current price. So for 65-inch and 75-inch TV, the price is different. However, for quantum dot LED that was manufactured by Samsung, the price is relatively cheaper, and you get the best quality as well. Yeah, so up to this much was the brief introduction kind of for quantum dots and nanomaterials. So from now I will be talking about, okay, for 20 minutes. So from now I'll be talking about perovskite, lead halide, perovskite quantum dots, also called nanocrystals. And this is a wonder material, and I have been working on it for like 4 years. And these materials are represented by ABX3 is the general formulas, and these materials are very inexpensive, and we can solution processable at low temperature. It has the high electronic properties, charge transfer properties, and when we use it for solar cell, it has high solar conversion efficiency. So ABX3. So A is like metal cation, like cesium. It has +1 charge, or it could be a small organic group like methyl ammonium or formamidinium. And B is like normally it is lead or tin, and the X is a halide ion. Some examples are like methyl ammonium iode, methyl ammonium lead fluoride, bromide, and formamidinium lead iodide. So here also, if you change the composition, suppose for the chloride, it emits around 400 to 450, and if it is iodide, it gives you red emission color. And this is a comparison of organic lead LED versus perovskite LED. So perovskite LED, it emerged in the year of 2014, and within very short time, it has reached a efficiency of about 20%. The organic LEDs were already in the market, and it has been for a long time, like since 1990s, and it has reached above 25%. So QD LED are also in the market for like 1995 to 1995, and it has reached efficiency above 20%. However, the perovskite LED introduced just few years back, and within very short span of time, it has reached a higher efficiency of about 20%. This is a room temperature synthesis where we the precursor in solvent like DMF or DMSO, and we spin coat it on a substrate and then anneal it. Then it gives different colors. So you tune the emission, you tune the concentration of the precursor material, and you get different color. Like yeah. Okay. This one is the room temperature synthesis, and this is the hot injection synthesis. So these are some syntheses done by me in our NTU lab. So here I have synthesized like cesium, lead, chloride, and bromide. The pictures on the right are under UV lamp. So this is cesium chloride, bromide, and this is cesium lead iodide. So depending on the composition, that is the halide ion, the color is different. And as we know, the ionic lattice also expands, and therefore we get because the size of the crystal lattice, we get different emission color. And the synthesis of this hot injection, we call it, because we inject the precursor at 180 degree in a nitrogen environment. So it involves 3 steps normally. So first step is the cesium carbonate, and you react with oleic acid, and it forms cesium oleate. And so cesium oleate, we inject it to the second reaction system, that is lead bromide. The lead bromide salt we dissolve in oleic acid and olefin, and when it dissolves at 120 or 130 degree, and we raise the temperature, and then in the second step at 180 degree, we inject this one, the cesium oleate. So it first forms a complex of like lead bromide oleate kind of, and then to that we add cesium oleate, and then it forms cesium lead bromide. So suppose if you want iodide, then you use lead iodide, or if you want fluoride, then you change the precursor here, the lead, whether you use lead chloride or bromide or iodide, and you get different color. So this is one of my recent article that, and here we made these nanoparticles like cesium lead bromide. The middle one is a green one that was synthesized by using this method, and then we added different precursor to tune the color. So first we synthesize the green one, and then we make different. So here is this method is called the halide exchange method. So what we do, we have the nanoparticles, the cesium lead bromide nanoparticles in toluene, and as we know that toluene and water is immiscible, so we add this. This is a salt. Formamidinium iodide is a iodide salt. So we add this salt in water, and then we add it to the nanoparticles, the nanocrystals that are in toluene. So water and toluene, it will form 2 different layers, and the iodine will go into the nanoparticle, and it will form the it forms the red color. And then we separate the water, like in the bottom we have the water, and in the top layer we have toluene. So we separate the 2 layers. And we found that when we make nanoparticles by this method, like halide exchange, first you make the bromide nanoparticles, and then you go for halide exchange, like from bromide you go to iodide, then you form very stable nanoparticles. So we did some NMR characterization to confirm that only the iodide ion is going inside the cesium lead bromide, and formamidinium part is not going inside. For that we have done some NMR spectroscopy and also XRD. So XRD confirms that we have iodide. So because when iodide goes into the crystal lattice of bromide, cesium lead bromide, the peak shifts. So the black one is for cesium lead bromide, and as we introduce iodide, there's a shift in the peak position. So this confirms that iodine is going inside. So the bromide one emits around like 510, and as we increase the concentration of iodine, the peak also shift towards the red. And these nanoparticles, these nanocrystals are like 8 to 9 nanometer in size. So we one very obvious question is that why we do halide exchange, and why don't we go for the direct synthesis? Like the direct synthesis means hot injection synthesis. So we found that we compared like the halide exchange are much more stable. However, if we do the direct synthesis, we see some impurity peaks. So it degrades, like you can see some yellow portions come yellowing on the film is happening, and also in solution also it becomes white color. It degrades to a non-emissive phase within very short time. However, the halide exchange ones are very stable, and we use these nanocrystals for making LED devices. So here we have used PEDOT PSS as a whole transporting layer, and POT2T is the is the electron transporting layer, and we have used an emissive layer as this quantum dots we have used as an emissive layer. So when we made devices, we found that our pure cesium lead iodide nanocrystals gives the higher efficiency of about 1.86%. However, the bromide iodide containing the mixture one is less efficient, and it has emission electroluminescence at 658, 663, and 670. And these are some LED, the color emission stability, like the curve represented by the green is the emission for the pure one, cesium lead iodide, that has emission at 668 nanometer emission. So this one is more stable, like you apply different voltages from 2.5 to up to 8, and the pure one, the cesium lead iodide, is more stable. Also, we checked different halide sources. Like before, we have used from ammonium iodide. So this time we use like different precursor like methyl ammonium iodide or tetrabutyl ammonium iodide, and it works perfectly. Also, we use some fluoride precursor, like from green we can go to red. Can we go to the blue side? So this also works perfectly. So we add tetrabutyl ammonium fluoride, and it the color can be tuned from green to blue. This is another work that was where we have done demonstrated some. I have 10 minutes. So here we have done some tin doping from ammonium lead iodide nanocrystals, and we have made NIR LED. So you know NIR LED, one very common example is the pulse oximeter, and we have been using it more during this pandemic, like what is the oxygen level. So you know this pulse oximetry is based on a principle that oxygenated hemoglobin, it absorbs near infrared light, it absorbs more than the hemoglobin. So depending also, this kind of oximetry is also it uses dual LED that has emission at around 761 and 881. And also near infrared LEDs are used for red light therapy, and also some other examples are like remote control, or even in the defense camera, those CCTV cameras, they also have a near infrared LED. So this perovskite quantum dots, we wanted to make some NIR LED, and because the stability is not so good, so we wanted to improve it by doping with tin. This paper was published last year in 2019. So here we doped different percentage of tin and tune the emission color from 797 to 775. So if we have more doping of tin, the peak shift to towards the from 97 to 775. So what happens on doping, that if we have less doping, then we get some with higher doping, the stability increases, you can say, and we characterize it till 3 months of synthesis, and the concentration of tin was determined by XPS. And it says that it has tin in 2-class oxidation state, and we made the LED devices using this tin doped one, and we got efficiency of around 1.7% and 1.4% of LED efficiency, and it has electroluminescence peak at 798 and 774 nanometer. And this is another work that was published from King Abdullah University in Saudi. So here we have made some cesium lead bromide nanoparticles, and we doped it with bismuth. So why bismuth is because bismuth and lead, the ionic radius is similar, and they have similar electronic configuration, and we dope the toxic metal. So we wanted to do inserting some bismuth ion will make it less toxic, and as well as bismuth is a heterovalent ion, like the lead is 2+ and bismuth is 3+. So this is a heterovalent doping. So here we start with the lead bromide and bismuth bromide salt together, and then we add the ligands, the oleic acid and oleamine. We dissolve it at 120 degree, and then inject the cesium oleate at 180 degree. So 165 degrees. So depending on the concentration of the dopant, the color, this is under visible light, the color is also different. And then we did some XPS to determine, like, although we have used like 5% or 10% of doping, but what is the actual concentration of bismuth that is going inside? So we determined, we found that the actual concentration, when suppose we are using 5% of bismuth, however, the actual percentage is only responding to because this XPS peak confirms that bismuth was actually inserted. And these are the TM images that says like the lattice spacing and the particle size didn't change when we incorporate bismuth. And also these are the UV and PL, the emission spectra. So we found that when we dope with higher percentage of bismuth, the emission intensity decreases, the PLQI, the quantum yield decreases if we have more percentage of bismuth. However, we did some charge carrier dynamics, and we found that the bismuth doped quantum dots are better in charge transfer. This is another work from NTU where we have done some partial kind of work. Here we have the cesium lead bromide. We capped it with some silanes, and it is like a partial structure. And this is one work that was published in the Nature Communication. So here also we made different sizes ranging from 7.5 to 12 nanometer in size. And these nanoparticles, the size is different because we have used different ligand. The capping ligand is different, and also we use different centrifugation speed to isolate different batches of nanoparticles. And the emission also changes from 746 to 800 or 807 for the film. So depending on the size, the emission is different, and we used it for measuring multiple exciton generation. So multiple exciton generation, it is a method where, like I have in the beginning I said, like exciton, the electron hole pair. So these quantum dots, they have the property that it can generate multiple exciton. Like normally when it absorbs a photon of light, it will give you a single pair. So suppose these quantum dots will give you a multiple electron hole pair. So more the charges means you create more and you create more electron hole pair. So normally the conventional quantum dots like the lead sulfide or the lead selenide, they generate because they have very high quantum confinement. So it is not so easy to isolate the and they suffer from like a phenomenon called hot carrier cooling. Therefore, however, these quantum dots, the lead halide quantum dots, they are just perfect for the for this multiple exciton generation. And we found that the intermediate size, that is the 8 nanometer, not the very small one, not the very big one, but the intermediate one gives better efficiency. Okay, so during my PhD work, I have worked on zinc sulfide quantum dots and doped with different metal ions like manganese and copper, and also double doping like copper and manganese. So this work where we have used copper doped zinc sulfide, and we used chitosan or chitosan also some people call it. So this is a biopolymer, and we made the nanoparticles. So initially the nanoparticles are like non-emissive. It was not showing any color. But when we add some reducing agent, like initially the copper 2, copper 2 is non-emissive, but when we add some reducing agent, it becomes copper 1, and then they're emitting some green color. So initially it was giving the emission color of the host, that is a zinc sulfide. It has emission around 420. And then when we add sodium borohydride or like vitamin C ascorbic acid is also a reducing agent. So this new peaks arises when we add when we add the reducing agent. And similarly, we can, after we form this one, copper one, we can again oxidize it and can kill the emission. So this the copper presence was confirmed by XPS. This is schematic representation where we added, initially it was like a piece like this. But when we add tripolyphosphate, it breaks this cytosine and it forms a composite nanoparticle. And then to this one, we added sodium borohydride, and this composite nanoparticle are also emitting. So this composite nanoparticle, for this one, the size is around 100 nanometer. And yeah. And then we use this composite nanoparticle for bioimaging purpose. So under the microscope, it looks like this. And when we, these are some biological cells, that is HeLa cell is a cancer cell, and this is embryonic cell. So when we incubate the biological cell with our quantum dots, with our copper dot zinc sulfide quantum dots, they emit very bright green color. And we used a reverse reaction, that is adding some pyrogalol to kill the emission. So in that case also we see the emission is vanishing. And we perform this XTT-based cell viability test to check that if our nanoparticles are like, are they favorable, like when they go inside the cell, are they killing the cells? So we found that like almost 100% are still living even after treating the biological cells with the quantum dots. So both these cells are like HeLa cell. The first one is for HeLa cell and second one is for HEK cells. Both were surviving even after incubating with our nanoparticles. So this is a schematic representation. This is another work where we have doped with manganese and copper. So here also we tune the color depending on the like whether it is oxidizing environment or reducing environment. We can change the color from orange to yellow or to blue. In an oxidizing environment, it emits blue, and in a reducing environment, it emits yellow. So we have done some characterization by emission and UV spectra, and this is the schematic representation for the energy levels. We have tuned these colors. This is my latest work that is going on in NTU. I have a very short film to show here. Okay, so this is octyl ammonium tin iodide. So this one I recorded yesterday just for this presentation, and this is very interesting. This is in ice water. So this is octyl ammonium tin iodide. So tin iodide is red emitting, under UV light it emits red color. So we do the synthesis and then dip it in the ice water bath, and it becomes like red blood color. So similar, this is for iodide, like octyl ammonium tin iodide. However, if it is bromide, then it emits blue color. Okay. Okay, this is my final slide. Okay, so like quantum dots are not so new. If we say like, you know, they were introduced in the year 1900, early 1980s. But still, we are trying to improve their synthesis method. We are trying to improve their properties so that they work actually in the real world, and we collaborate with other departments as well. Thank you. I think I haven't checked the chat window yet.
Okay. Thank you very much, Dr. Raihana, for interesting and comprehensive presentations. For participants, if you have any questions, you can type on chat box or directly by raising your hand. you can ask in Bahasa Indonesia, and we will try our best to translate into English.
Would you mind describing, this is from Halida. Would you mind describing about doping agent type role in semiconductor nanocrystals in order to eliminate doping agent type role in semiconductor nanocrystals in order to eliminate surface defects? I don't think the agent type role. A type role, if you mean to say that whether like sometime when you're doping the doping agent, the doping ion, that is suppose manganese or copper, sometimes it goes inside or sometimes it remains on the surface. So the one that goes inside the crystal, that is they contributes to the emission. And those that are loosely bonded on the surface of the nanoparticle, they normally act as a defect. So sometimes removing those excess ions or like the optimum concentration is better to get the maximum emission and as well as reduce the surface defects. Another in the application of quantum dot LED, recognizes weakness in dark light and can be doped with other material. What are the transform? Dark light. That I do not have any idea. Dark light and can be doped with other materials. I do not have any idea. Okay, is there, the questions are mostly regarding the registration and
yeah, sorry for that. Dr. Rayhana, I have questions. Is it possible to make quantum dots from any material? Yes, in your case, you create quantum dots.
Not any material.
Yes.
Yeah. So like in the beginning, I showed like you need to have only like zinc and then sulfide,
yes,
or like only particular groups of the periodic table only works. It's not like a random picking. Yeah, so like 3,5, like group 3 indium phosphide, those kind of combinations only work. However, the carbon dots are like some recent reports, they make carbon dots from even biscuit, or that was from my previous group, IIT Guwahati, where they made some from cornflakes, they made carbon nanoparticles, or from biscuit, those kind of things. So carbon dots, yes, from any organic source, we can make not any, I mean some.
I see.
But chalcogenide quantum dots, mostly they are from particular groups of the periodic table. Would you mind describing about doping in the application? Quantum dot application is very good, but how do Indonesia, did the synthesis is expensive? No, hot injection synthesis is expensive. Suppose for the perovskite nanoparticles that we do in NTU, the hot injection synthesis is difficult. Like first you do the reaction in a vacuum condition. So you need a vacuum pump, you need a fume hood to do this reaction, you need nitrogen environment. However, the low temperature, the LARP method, that can be done, that is not expensive. You just need a precursors and you dissolve it in isopropanol or some polar solvent and then add it to toluene. So even that experiment can be introduced in master degree syllabus for regular experiment. Is very simple. And also, I think those synthesis of gold nanoparticles, silver nanoparticles, those are also very simple to synthesize. What is the surface specification and what is the function under research? So surface specification, as I said, like the nanoparticles has the tendency to go become bigger. Like nanoparticles, they have higher surface energy, they have higher surface area. So this is a natural tendency, like they want to get bigger, they want to get agglomeration, they want to form some defects. So we want to do the surface specification, like you cap it with some organic molecules so that the nanoparticles are like distance, one is here, one is here, not like you don't put it together. they want to come closer, but you do some surface specification so that they are like they're far kind of. So you do the surface specification, and also surface specification you do because you want to do some, you want to solubilize in some solvent, like in water or in some hexane kind of like non-polar solvent. And suppose it is, and also because those defects, they kill the emission, they try to reduce the emission intensity. So when you cap it with a ligand, you do surface specification, it increases the quantum yield. Yeah. How to simple test quantum dot particle size? So quantum dot particle size is mostly determined by TEM, transmission electron microscopic images. You got that one. That is the most accurate method. And otherwise, there is one dynamic light scattering, DLS method. So that also gives you an average of like what is the size range. Did Dr. Noel journal the extent of? Okay, I don't know. Okay, is there any question?
Okay, I think time is up. Thank you very much, Dr. Rayhana. There are so many questions from our participants. I think your presentation is so interesting for them. Okay, once more, thank you very much, Dr. Rayhana. Okay.
Thank you so much for giving me this opportunity.
You're welcome. Okay, we see you next time. Thank you.
Okay, thank you. Thank you so much for this opportunity to speak before the students. I hope to present sometime in person coming to your university. Thank you.
Okay, we're
so
waiting
much.
for you.
So I will leave it. I leave the session.
Okay.
So I will leave it now, right? Okay. Thank you.
Okay, no problem.
Or is the next session going on?
Yes, we have the next session by Professor Eric Harris from Groningen University.
Okay, I will attend it. Yeah. Okay.
See you. Okay, before we move to the next speaker, we will play a video profile from the chemistry department of Riau University.
Universitas Riau is one of public university located in Pekanbaru. Founded on October 1st, 1962, Universitas Riau is celebrating its birthday this month with various exciting events, including this international webinar. Natural Science are one of the 10 faculties in Universitas Riau. Department of Chemistry is one of 5 departments under this faculty. Department of Chemistry has undergraduate, magister, and doctorate program. Both of undergraduate and magister program has received A accreditation from Indonesia's National Accreditation Board for Higher Education. Department of Chemistry of Universitas Riau aims to create high quality and competitive graduates in Southeast Asia on 2035 and to be one of the best research center in both life and non-life natural resources. Our research focused on 5 key areas. First is catalytic conversion on biomass and waste treatment, led by Professor Amir Awaludin, Dr. Nur Hayati, Dr. Amelia Linggawati, and Dr. Leo Saputra. Bioactive compounds from local biodiversities, led by Professor Titania Nugroho, Professor Saryono, Dr. Hilwan Yuda Teruna, Dr. Rudi Hendra, Dr. Yohana Nurulita, Silvera Devi, Andi Dahliati, Yuli Haryani, Dri Novianti, and Yohana. Thirdly is drug design and organic synthesis, led by Professor Adil Zamri, Professor Jasril, and Dr. Yum Erianti. Fourthly is petroleum and biofuel chemistry, led by Dr. Sofia Anita and M. Rizal Mahidin Tambusai. Is adsorbent, coagulant, and biocomposite, led by Dr. Mudarina, Halida Sofia, Ganisvia Kartika, Pepi Halsayanti, Tengku Ariful Amri, Tengku Abu Hanifah, Itnawita, and Muklis. Our students has received numerous awards, such as funded research by Ministry of Education, Novice runner-up of NUDC 2019, second runner-up of National Chemistry Olympiad by MIPA Expo, winners in some paper competitions, and many more to come. We are so honored and thrilled to welcome you on board with us.
Okay, thank you very much for watching the video. Our last speaker of this webinar is Professor Dr. Hiro Jan Hiris from the University of Groningen, the Netherlands. Okay. Prof. Hiris is a professor in green chemical reaction engineering at the University of Groningen, the Netherlands. He performed a postdoc at the University of Oxford on asymmetric catalysis in 1990. His research interests concern the development of efficient catalytic technology for acid and metal-based catalytic biomass conversions, with an emphasis on biofuels, platform chemicals such as lefulinic acid or hydroxymethyl furfural. Prof. Hiris is author of more than 250 papers and 12 patents, with a citation score of 14,797, and Prof. Hiris is a member of Royal Holland Society of Science and Humanities. He is also an associate editor of the journal Fuel Processing Technology and editorial board member of the journal Catalyst. His topic for this webinar is green energy harvesting from biomass. Okay, Prof. Hiris.
Thank you very much.
Could you hear me?
Can you hear me? Can you hear me? Can you hear me?
Yes, I can.
Oh, okay.
Very good. So I will share my screen.
Time is yours.
Okay, thank you very much for your kind introduction. Can everybody see my screen? Is the technique working?
Yes, I can.
Thank you very much. First of all, I'd like to thank, of course, the organizers of this webinar very much for allowing me to speak here. I have been in Indonesia many times, and actually I've also been in Pekanbaru, I think about 5 or 6 years ago, and already gave a presentation there at your department. And actually also I'm very pleased that Professor Amir also once visited our university in the Netherlands already, I think 10 years ago, and we all at that time had a very nice collaboration and very good discussions. Now, what I will talk about today is a topic from biomass to bio-based products, and energy will be one of the aspects I will cover. Now, first of all, some of you may think, where is Groningen? Huh? Now, I put it on this slide. Here you see the Europe, here you see the UK, here you see France, here you see Germany, and the Netherlands is a tiny dot. We're about 18 million people. Here you see the capital of the Netherlands, Amsterdam, and here you see Groningen. It's actually in the north, close to Germany, and our university has about 32,000 students. Here you see a picture of one of my colleagues. and his name is Professor Ben Veringa, and we are very proud of him because he won a Nobel Prize for Chemistry now 4 years ago, and he's
We
one
are
of
sorry,
our.
we need to wait response from Professor Hess.
Sorry, can you repeat? Can I continue with my presentation?
Yes, sir, please.
The content of my presentation will be the following. First of all, I will give a short introduction to the topic. Why should we use biomass? And what are the options we have if we use biomass? I will then focus on a particular technology, which is called pyrolysis. And I will show you some of the possibilities we have when we pyrolyze biomass into products. Then I will move a bit more to the production of chemicals, renewable chemicals, and give you an example there. I will particularly focus on aromatic chemicals from biomass. I'll end up with some conclusions and then finally an acknowledgement. I hope you see this picture here, and you may think, what is this? Is this a part of a textile, or is this cotton or something like that which has been stained? No, actually this is wood, stained wood. So this is the internal and this is the outside of the wood, and you see here the structure of wood. And apologize, wood is a very important starting material when we actually use biomass. Now, why should we use biomass? What's the incentive for that? And I hope we all know that at the moment, most of our products, and particularly transportation fuels and also chemicals, are made from fossil resources. And we all know the issues related to the use of fossil resources. So there is actually a large incentive to actually move away from the use of fossil resources and actually move into a world where we use biomass also as a source. And an economy where biomass plays a major role is what we call a bio-based economy. And a bio-based economy is a future society where we're not only depending on fossil fuels or fossil resources, but we also use renewable feedstock like biomass to move away from a conventional oil well, and then move into a situation where we use more agricultural residues, for instance, for all kinds of applications. Now, what can we do with biomass? And particularly, this is the starting point. And here we see some products we can derive from biomass. We can go for a low value application, just burn that biomass to heat and power. We can move up a bit and go for transportation fuels. We can also move to bio-based chemicals and then move up a bit further and we move to bio-based performance materials. To go from biomass to these product fields, we need technology. And here you see a number of technologies in this block here, which we can use actually to do something with biomass. And here I have discriminated in a high temperature platform and a low temperature platform. What do I mean with this? This is roughly, let's say, 400 degrees plus, and this is below 400 degrees. Now, what you have to think about is we talk about high temperature, think about biomass combustion, biomass gasification to synthesis gas, pyrolysis, which I will actually go into detail in my presentation, and then finally hydrothermal liquefaction. And when considering the low temperature platform, you can consider pretreatment, hydrolysis, fermentation, for instance, to bioethanol, or you can separate in fractions and then use these fractions for these applications. Now, often the primary products from these conversion technologies are not directly suitable in these applications, and you have to do secondary conversions. You have to do something with these primary outlets to convert it into these products. Now, for those of you who, what I particularly focus on in my presentation is non-food biomass. And the reason is, I think, obvious. We would like to go for a biomass when there's no competition with food. So we would like to aim for biomasses which are particularly woody, of a woody type. And you can think about straw, you can think about residues from the wood industry. You can think about energy crops like miscanthus here. And these could be non-food biomasses, second generation biomasses, which we can use for all kinds of applications. Now, what I'm working in this field here, has been said before already, a long time, about 20 years. And the incentive is then, of course, that we move away from a conventional oil refinery, which is schematically given here, where we move in a situation where we biorefine our biomass, and biorefining means that we convert the biomass as efficient as possible into useful products like fuels, electricity, and materials. Then we do that with a short CO2 loop. And what is also very important, that we also recycle the products we obtain. So the recycle loop is also important in future development. And of course, biomass contains ash. So we also should be very careful about what we do with the ash and the minerals, which are often present in a number of biomass sources. To make this transition happen, we require new catalysts, we require new technology, and we have to develop new processes. I think that's one of the important tasks we have to fulfill to make this transition from a fossil-based economy to a bio-based economy efficiently. And so there's a lot of research activity going on to make this happen at the moment. Now, second part, we will have a look at pyrolysis. I've shown you that pyrolysis is one of the biomass conversion technologies. What entails pyrolysis? Once here, so I will focus on pyrolysis. Now, the principle for pyrolysis is actually very simple. It's actually heating up the biomass, schematically given here, heating up in the absence of oxygen, and then what will happen is the biopolymers in the woods, they will break down, the cellulose will break down, the hemicellulose will break down, as well as the lignin. They break down in small fragments which are volatile. The volatiles you actually condense, and the condensables form a pyrolysis oil, and you also end up with some non-condensable gases. There's always some char formed, that's unavoidable, and actually this is not a waste product. We could also do something with the char. Now, typical conditions, temperature between 400 and 550 degrees. You need a short vapor residence time, so this process of cooling down the vapors has to be done very quickly. And what is also needed is a very high heating rate of your biomass particles, because then the yields of the oil are the highest as possible. Now, what are typical yields which are attainable? It's something, if you do this properly, you can actually roughly get between 60 and 70% of the original biomass. You can convert that into a pyrolysis oil. Is this still in a lab scale? No, it's not. It's actually been scaled up already to commercial scale. And this is a slide from a company called BTG. It's a company from the Netherlands, and they actually try to license and try to build pyrolysis units worldwide. And they now have successfully implemented some of this technology in real, actual commercial plants. And how does this look? Well, here you see again the biomass. The biomass is fed to the pyrolysis reactor. And the pyrolysis reactor is something very special. And actually the pyrolysis reactor actually is not only an empty tube, but what is in there is also sand. So you combine the biomass with hot sand, and here the pyrolysis takes place. The vapors, you condense, and you get the oil, and the byproduct, the sand and the char, are going out of the bottom. And this end up in what we call a kind of recycle loop, and where the char is also used. So it's not thrown away, it's used again. What's happening here is the sand and the char are fed to a combustor with air. There the char is burned off, it heats up the sand again, and the sand is fed back to the reactor. So the sand is used as the heating medium for the system, and actually the heat management is done by the char, which is also formed. Also from the combustor, you get some flue gases, and also here you get a non-condensable gases, and that you can also use for steam generation. Now, is this has some pictures of this commercial unit is given here. It's a unit called Empiro, and Empiro is built in Enschede, close to the German border, about 150 kilometers away from Groningen. It was erected in 2016. What are they doing? Well, there's a lot of information here. what is important is that they have a capacity of 5 ton-feet per hour, and they make about 3.2 tons per hour of pyrolysis liquids, and together with an amount of steam and also with the electricity. So this is a semi-commercial pyrolysis unit, which is actually in operation. Well, here you see an overview of the unit, how it's actually built. It's a modular system. Here you see the pyrolysis unit. Here you see the biomass feeding system, and here you see the oil collection. And this oil is actually used in a neighboring factory for actually generating steam in a boiler. Now, so you could ask, why do you want to transform biomass into a liquid, into a pyrolysis liquid? Why is this interesting? Well, be aware that, first of all, pyrolysis oil is a liquid form of biomass. So the energy content is much higher on a volumetric basis. That's one. Secondly, liquids are by far more easier to transport than solid. And finally, because you pyrolyze and you vaporize part of the products, and that means that the ash is often remaining in the char and in the sand. And so it's also the amount of impurities, minerals, are by far lower in this situation. The pyrolysis oil has of course properties, and be aware it's not a fossil crude oil. It's not a petroleum type oil. First of all, it contains a lot of water, still 15 to 20%. The pH is rather low, so it is corrosive. It contains, for instance, a bit of acetic acid, which you always get when you pyrolyze wood. It's a very complicated mixture of more than 400 components. Some of them have a low molecular weight, so you can actually see them by techniques like gas chromatography. Other ones are higher molecular weight, so there's still a high molecular weight fraction in the pyrolysis oil. And also, it contains still oxygen, because if you vaporize the biomass and you cut it down in smaller fragments, there's still oil, there's still oxygen, bound oxygen present, and this can be up to 25%. So it's not a crude oil. And finally, the storage stability is also a bit limited. It's up to several months, but when you store it too long, it starts to form deposits. And of course, this is also something you want to avoid. Now, what can we do then with the pyrolysis oil? You can actually use it for combined heat and power generation in a diesel engine. You have to modify the engine. A typical diesel engine doesn't work on pyrolysis oil, so you have to do some modification to make that happen. There have been tests in the past where it has been co-fired in power plants, and also actually one of the nice applications is you can use it actually to improve the quality of meat you put on a barbecue, and you can sell it, for instance, in the US. You can buy it in the US, and then it's called liquid smoke, and it's used to flavor meat before you barbecue it. But it's a very small application. Now, if we look to only pyrolysis oil, of course the question comes, can we do something more with the oil? Can we do something with the oil which gives it high value, and where we extend the application range? Yeah, there are opportunities, but then you have to do a secondary conversion. And that's what I'm showing in this slide. So if we start again with biomass, you can pyrolyze this, you get this pyrolysis oil with a limited number of applications. But what you can do then is give it a certain treatment, a catalytic hydrotreatment. And this is a treatment where you use a catalyst and hydrogen. I'll explain later in more detail. And when you do this, you can actually remove the bound oxygen to a certain extent. And when you actually remove all the oxygen with this treatment, you end up with a liquid product which is actually fully hydrocarbon. So it's more like a fossil or a diesel or a gasoline. And of course, this is advantageous. So then you go from biomass to a product which is diesel, gasoline alike, and may be used directly in a combustion engine. The other way, which is also an interesting one, is that you make what we call a stabilized pyrolysis oil. And in this case, not all the oxygen is removed, but you stabilize it in such a way that it does not form deposits when heating up and things like that. And then you enter in a very interesting application. What you can do then is actually co-feed this material in an existing refinery. So what you actually do then is you have an existing oil refinery, and somewhere in the refinery, you introduce a pyrolysis liquid, let's say on a blending rate of 10%, and then part of the outlet of the refinery contains green carbon, bio-based carbon. And so it's not a fully green product which you make here, but it's a partly green product by co-feeding in an existing refinery. Now, if we look to a refinery, an oil refinery, it's a very complicated process. It has been optimized over a period of centuries. I'm not going into detail in this, but here you see the crude oil coming in, and there's distillations at atmospheric pressure and other pressures. Then there are catalytic conversions, and it all is aiming to get as much gasoline, diesel, and jet fuel out of a refinery. And the question then came, where in this refinery can we introduce this improved quality pyrolysis oil? Now you can think about, okay, maybe we can add it here, and just before the atmospheric distillation. Just blend the crude oil with the pyrolysis oil and then see if this works. You can also do it here. This is the FCC unit. It's a unit where you crack heavier oil fractions into lighter ones. And this could also be a nice entry point for improved for pyrolysis oil. Another one could do it in the hydrotreater, where you actually remove sulfur and nitrogen impurities in the fossil resources. And that could also be an interesting option to introduce. Now, before saying something about this, I would like to first introduce you the process of catalytic hydrotreatment. So what's the idea here? We have a fast pyrolysis liquid. We contact it with hydrogen, molecular hydrogen, with a catalyst, suitable catalyst, at elevated pressure and temperatures. And what we would like to have is a high yield of an oil which is more stable, which is upgraded, and which we can use for various applications. Inherently, you always form water, and it's also unavoidable that you form some gas phase components, and like CO, CO2, etc. This is the chemistry which you would like to happen. And you have here the pyrolysis oil, schematically given here, which contains carbon, hydrogen, oxygen, bound oxygen. What you like to do is remove the oxygen by doing a reaction with hydrogen, and then form water, and then you end up is your hydrocarbon. This is the chemistry we would like to achieve. This is not that easy. It's typically high temperatures. These are the temperature range, and also you require high pressures. It's a bit similar to those of you who are familiar with terms like hydrodesulfurization or hydrodenitrification, and the catalyst type and also the chemistry is rather similar. Now, in our lab over the last 2 decades, we did a lot of work actually to identify good catalysts, which do the trick. And because you can probably imagine that the catalysts used in the petrochemical industry are not suitable for pyrolysis oil. Pyrolysis oil contains water, and water is always a problem for catalysts. So you have to develop new catalysts. Now, we actually were able to do so and characterize them in a lot of detail with all kinds of techniques. What we also did was, studies on a more reactor engineering level, and where we actually try to develop reactor configurations which are very efficient and which gives you a high yield of the desired products. And this is just one of the configurations we use. It's a continuous setup where we here feed our pyrolysis oil. We also feed hydrogen here. Here you see 4 reactors which are filled with our solid catalyst. You pass it over. Here the hydrotreatment takes place. You collect, you cool, and you collect the liquids over here, and you analyze it in detail, and you also collect the gas phase and also analyze that one in detail. Now, if you do a catalytic hydrotreatment, you can probably imagine that there are quite some requirements if you want to do this in a techno-economically viable way. So you want to develop cheap processes. From a process point of view, you don't want to use a lot of hydrogen. You need it, but you want to minimize the amount because hydrogen is expensive. You would like to have a high carbon yield, and you don't want to lose carbon in the form of gases or in the form of char. The catalysts have to be active, stable, and they should be preferably cheap. And they should not be based on palladium or rhodium and very expensive metal catalysts. And also you'd like to reduce the temperature and pressure as much as possible, because then also the price of the equipment will be lower. And also from a product point of view, you have quite some considerations you have to consider. For instance, you would like to reduce the oxygen content of the pyrolysis oil, but the exact amount depends on what you would like to do with the product. If you would like to co-feed it in a refinery, then you probably don't need an oxygen content of 0%. If you directly want to use it as a gasoline or diesel substitute, you need to go to an oxygen content of below 0.5%. Now, the viscosity should not be too high, should not contain water, the thermal stability should be good, and preferably it should be miscible with conventional hydrocarbons like diesel and gasoline. Now, we did a lot of work in this field. You see some older publications. We started with a benchmark catalyst, which is ruthenium on carbon, which does the job reasonably well, but is expensive. Then we ended up in bimetallic systems. Then we tried to use cheap catalysts, which are preferably nickel-based. And then we found out that particularly nickel-copper catalysts are very good and do the job excellent. And then we further improved them by tailoring the properties of the catalyst, by playing with the support. And then we developed Picula catalysts, which were actually patented also by us. And these are at the moment our best catalysts we can provide for the hydrotreatment of pyrolysis liquids. Now here you see then a picture, and what is a typical oil product after hydrotreatment. What you see here is a number of you see here a liquid phase, which is actually the product oil, and you see here the water phase, which is formed by the chemistry, and there's also still some organics in the water phase. Now, as I told you, we also do this on a continuous level. This is just some typical conditions we use. A hydrogen pressure about 200 bars, so still high. The weight hourly space velocity is 0.1 to 0.6 to 1 per hour, to give you a bit of an idea about the activity. And these are typical analysis we do on the oils to see whether they are within the range as we expect them. I don't have time to give you more details about this. We have obtained a lot of information about the chemistry which is going on during the hydrotreatment. We know a lot about which molecules cause problems when you do it and which you have to avoid and how to avoid, but that's beyond the scope of my presentation today. So the findings are we developed an improved catalyst, which we call Picula catalyst. In the company BTG, they have used these catalysts in advanced continuous setup and operated for more than 500 hours. So these catalysts are relatively stable. You can use them for a prolonged time on stream. We also saw that we can tune the oxygen content of these liquid oils. You can make them oxygen free, no problem at all. Then you run them with transportation fuel. And you can also stabilize them at between 5 and 20%. And then you can co-feed them in existing oil refineries. Now, we have done some tests in other projects, European Union projects, with where we co-feed the upgraded pyrolysis oil in FCC units, so hydrocracking units, and these were very successful. And actually now there are a number of companies, large petrochemical companies, they're doing research in how to implement this in a real oil refinery. Now, how do these oil look like? It's also interesting to see. Here you see a typical pyrolysis oil, and here you see the different fractions you can obtain after the hydrotreatment. And the more you go to the middle, the deeper the oxygen removal level is. And this is a product which contains a very low amount of oxygen. And actually, when you do the analysis, it resembles diesel and it resembles gasoline. Okay, so this was pyrolysis, where I would like to show you how we can use that for fuel generation, for gasoline and for diesel. Now I'd like to move on to how we can use also biomass for the synthesis of renewable chemicals. And why is this important? Well, particularly in the Netherlands, but also in Europe, there's strong policies going on at the moment. And what we'd like to achieve is CO2 neutral societies already in 2050. So that's a lot of very ambitious targets, and we have to do something about this. And particularly in the field of chemicals, renewable chemicals, we have a major challenge here because for chemicals, there are a limited number of sources available which we can use. They need to contain carbon. And the challenge we have here is how can we compete with fossil resources? Because this is, of course, very difficult. Fossil resources are very cheap and we have to compete with them, so we have to develop very efficient catalytic processes. Now, what approach can we follow if we want to make a carbon neutral chemicals? Now, here you see a number of technologies available. The first one is use of biomass, and that's where I will actually focus on in my presentation. The second one, of course, also an interesting one, is start with CO2. And CO2 contains also carbon. We have too much CO2 in the atmosphere, so we can actually use it. And you can even do double green, where you take CO2 gases from biomass conversion plants and then take that CO2 and use that again to make chemicals. And there's another option where you actually use electricity when possible, for instance, when generating steam as auxiliary materials inside existing chemical processes. Now, I would like to give you an example. There are many other possibilities. Many studies are going on, and many chemicals can be made from biomass, but I would like to give you an example where we produce aromatics from biomass. So what's the idea here? We would like to make bio-based aromatics. And what are aromatics? Aromatics are BTX, benzene, toluene, xylene. So here, this is the molecules we are aiming at. Benzene, toluene, and the 3 isomers of xylenes. And we would like to make them from biomass, and particularly, preferably cheap biomass sources. And you can think about agricultural residues, wood residues, but also residues from the pulp and paper industry. And for instance, this is black liquor. And black liquor is a very cheap source and which we can actually use. Now, the idea is then we'll take the biomass, we turn that into these bio-based chemicals, and then convert them into interesting polymers. The interesting, when you focus on bio-based chemicals, bio-based aromatics, is that these are existing molecules. They're made already on large scale in the fossil industry. They are already the building blocks for polymers at the moment. The only thing we would like to change, that instead of using fossil resources, we would like to use biomass. And the advantage when you make an existing chemical is you can make use of the existing infrastructure to make this happen. And then the whole introduction of this technology will be more easy when you actually when you make a totally new product. So we use existing infrastructure. The only difference is we use biomass instead of fossil resources. Now, why did we select BTX? Now, because there are many companies making these aromatics at the moment, major petrochemical industries who do that, and they would like to green up their businesses. So they're looking for ways to make these compounds green from biomass. A well-known example is Huntsman, and we collaborated with them, and Huntsman wants to green up the benzene business. What is also important is that these aromatics now from fossil resources, they're used to make polymers. And what we actually see is that the polymer industry will grow dramatically in the future. And so it will be highly advantageous to replace fossil carbon by green carbon. And I would like to illustrate this by this picture. Here you see the predictions for the growth in plastics consumption on a global scale. And this is I like this graph very much. On this axis, you see the GDP, the gross domestic product per capita in thousands of dollars. And here you see the consumption of plastics. It's a relatively old picture, but it's still valid. So what you see when highly developed countries like the US, with a high GDP, they have a relatively high consumption of plastics. And you see there's a relation between the GDP and actually the plastic consumption. Now, we all know the GDP in the coming decades will increase. We will move in this direction. And this will imply that all the countries which are here now will start to use more plastics. So based on this rather relatively simple graph, we can predict that the amount of plastics which will be produced in the future will grow exponentially. And so there will be a large need to make them green. Now, what is one of the technologies we can actually use to make benzene, toluene, and xylene? And that's not pyrolysis, but it is catalytic pyrolysis. And what we do then is exactly the same. We have our biomass source, we pyrolyze it, we get a gas, we get a liquid, and we get a residue. But now we do this in the presence of a catalyst. And a well-known catalyst are zeolites. This can be natural or they can be synthesized, like H705. And when you combine the two, you're actually able to convert the liquids into a product which contains a lot of benzene, toluene, and xylene. Now, how does this work? This is a graph you've seen before. This is how we can make pyrolysis liquids. But now instead of making pyrolysis liquids, we would like this oil to contain a lot of benzene, toluene, and xylene. And what we have to do then is add a catalyst. And we can either do it here in the reactor, or we can do it here in the vapor stream, where we actually take the vapor and do the aromatization and the formation of benzene, toluene, and xylene in the vapor phase. This is what we call in situ. This is what we call ex situ catalytic pyrolysis. We prefer this approach, ex situ catalytic pyrolysis. So everything I will show you is based on this approach. Why? Because if we use the in situ approach, we always have the problem that biomass contains ash. Ash in combination, minerals in combination with zeolite catalyst is not the best combination, and it will lead to actually a deactivation of the catalyst, and that's something you don't want. Now, what are the let's say the challenges in this field to make BTX from biomass? First of all, we need to have cheap biomass sources. Secondly, we have to find ways to increase the yields of BTX by choosing proper reaction conditions, by further catalyst development. They should be robust and stable and active, and by using recycle concepts. And what is also needed, this is still on a level of lab scale. We have to go to higher TRL technology levels. So this technology also has to be developed on a pilot scale and then later on semi-commercial scale. Now, we do this on a lab scale, very small scale, where we do our catalytic pyrolysis, and we do this in a tandem microreactor. And in this tandem microreactor, it looks like this. It's here we have a gas flow. This is the pyrolysis unit, where we can introduce the biomass source by injecting it in the pyrolysis chamber. Then the gas flow takes the vapors and transport it to the reactor zone. And here the catalyst is present, and the chemistry takes place. And then the vapor outlet is connected to a GC. And actually, we can then directly see how much product we make and which products we make. And this is then a typical GC plot you get from this tandem microreactor. And here you see the benzene, here you see the toluene, here you see the xylenes, and here you see some other higher aromatics. Now, these are some typical conditions. It doesn't matter that much. For those who are interested, and you can have a look at, for instance, this paper, which we published relatively recently on this topic. I will just give some examples. We did a lot of work with wood. We did a lot of work with lignin. We did a lot of work with the residues from the paper and pulp industry. And we also worked on glycerol, crude glycerol. And you may say, why? Why would you like to use crude glycerol? Now, the reason for this is it's a waste product from the biodiesel industry. And what would be nice if we can take a green residue and convert that into benzene, toluene, and xylene. So we did a lot of work with crude glycerol. And actually, when you do the catalytic pyrolysis with crude glycerol, you get this product package. About 35% of an oil, water, which is inherent, some solids, and also some non-condensable gases. And when you look to the liquid, that's what we're interested in, the benzene, toluene, and xylene, about half of it is BTX, and about the other half is higher aromatics. So this is quite interesting. So we can make with crude glycerol about 78% of BTX. And here you see actually the distribution of the benzene, toluene, and xylene. So we mainly make toluene. Now, then we also thought about, okay, we also have these higher aromatics. And can we also convert them into these benzene, toluene, and xylene? And then indeed, this also works. So we do then, we have the crude glycerol, we do this catalytic pyrolysis, we make aromatics, we do a distillation where we get the benzene, toluene, and xylene. What is left are these higher aromatics. We treat them and we again recycle them back to the reactor. And in that way, we're able to also convert these higher aromatics and finally ending up in, let's say, 35 weight% of BTX on our crude glycerol stream, which is a very nice yield when you consider the techno-economic evaluations. Now, as I told you, we do this on milligram scale. In our lab, we also have a reactor for catalytic pyrolysis on a scale of about 0.5 kilogram per hour. This is an auger type of reactor. You can see it in our lab. And now with a company called BioBTX, we also scaled it up to about 10 kilograms per hour. And here you see the unit, which is now on operation on our campus in Groningen. We can even go a bit further. The idea will be that we also convert this benzene toluene into interesting plastics. And that's what we also did in another project. Here we have the glycerine, the crude glycerol. We do the catalytic pyrolysis, and we actually get here the compounds. In this case, we're particularly interested in paraxylene, so only this one. We oxidize it to phthalic acid, and then do an acidification, and then react it with green ethylene glycol, and where we end up is PET, polyethylene terephthalate. And this is actually a material Coca-Cola also used, for instance, to make their bottles. And so what we can achieve here by doing this sequence, by having green paraxylene, by using green ethylene glycol, we can make a fully green PET, 100% green carbon. And this is what we did in our lab, and we're the first one to make this 100% green PET. This is the material, and actually we made some, not bottles, we didn't have enough material, but we made these small caps where you can put in cosmetics, and the top part here is a fully 100% green, transparent PET. And we're actually very proud about that. Now, the conclusion of my presentation, I think I've shown you that biomass has a potential to green up the petrochemical industry by going to green chemicals, but also going to green diesel and green gasoline. Bioaromatics are a very interesting key target molecule, and catalytic pyrolysis is a very nice way to obtain these. And I hope also, and that's also what I always try to convince and convey to my students, and that research on biomass is really inspiring and it's also a lot of fun. And of course, I would like to end with my acknowledgement. I'd like to thank a lot of people. Here you see the companies and also the funding offices which were involved. Here you see the people who are involved in the company BioBTX, and you see the pilot plant here on our campus. And here we see a group picture of our chemical engineering department and all the, not the students, but most of the PhDs and the postdocs and the staff. This was in an outing about 1.5 years ago. And I would like to thank you very much for your kind attention.
Okay, what a clear and interesting presentation. Thank you very much, Prof. Eric Harris. For participants, you can type your question on the chat box or directly by raising your hand. Okay, Professor Amir Awaluddin here. Professor Amir, do you have questions?
Okay, I want to ask to the Eric. Very good presentation, Eric. Very nice presentation. Pak Leo,
thank you.
You can hear my voice already?
Yes.
Okay. In the last presentation, you mentioned about how to convert crude glycerol into BTX, benzene, toluene, and xylene. And then you mentioned about the catalytic pyrolysis. What is the properties of the catalyst that you really needed in order to get the best yield for your process?
Yeah. So actually the basic catalyst we use is H705, which is a well-known solid acid catalyst. It's a zeolite material. It's available on large scale because also used in the petrochemical industry. And that catalyst already gives good yields. You can further improve the yields by modification of these catalysts. And well-known modifications are, for instance, the introduction of a bit of gallium or the introduction of a bit of zinc. And then actually the catalyst become a bit better and you can increase the yield. The main problem with these catalysts is that during reaction, you also get coke formation. So the catalyst also form coke. And what we actually showed in a number of publications is that the coke you can actually remove by giving it a treatment with air, and then you can recycle back the catalyst to the reactor, and then it does its job very perfectly again. And you can recycle it many, many times without a tremendous loss in catalytic performance.
So you are using the zeolite, right?
Yes.
Zeolite catalyst. Is it possible also to use transition metal oxide porous catalyst for your reaction? Like, for example, manganese oxide, like a porous material also, 3-dimension porous material for your process.
Yeah, we never tried this, but if you look to the literature for catalytic pyrolysis, you will see a lot of catalysts that have been tested for all types of biomasses, and the best ones are still the H705. So what you need is acidic sites, and you also need some Brunstad sites, and that gives you the best catalyst. Whether a manganese oxide, whether it works fine or not, I'm not aware about that. I would have to look it up.
Okay, thank you very much, Eric. Nice to meet you.
Thank you.
Okay, thank you very much. We have 2 more questions from the participants. Okay, the first question is from Esther Teo from Riau University. Okay. The question is, can PAH from other sources also could be recycled, like the PAH from glycerol in this system?
A very interesting question. What we said is if you recycle the PAH from the products you make from glycerol pyrolysis, can we also do it with other PAH? Yes, in principle, that's possible. So if you have a cheap, let's say, PAH stream, preferably from a biomass source, you can indeed recycle it back to the glycerol system, and in that way increase the yield of BTX. Yes.
Thank you. The second question from Siti Mandalika from Riau University. Biomass absorbs color waves. What if pyrolysis is carried out to regenerate biomass? Will chloroxide and carbon dioxide compound form? If so, how to solve it? If not, how to pyrolysis? The question, could it be understood, Prof. Eric?
I will try, because I'm not 100% sure what is meant here.
I'm not sure what. Okay.
Well, let me just say some general things. In general, a pyrolysis reactor is an all-eater. You can pyrolyze not only biomass, you can also pyrolyze plastics, which is also a very hot topic at the moment, and to pyrolyze waste plastics, and then actually obtain also a hydrocarbon type of liquid, which you can either use for energy generation. You can also maybe use for converting gasoline and diesel. So a pyrolysis unit is an all-eater. And of course, you have to be careful when you feed sources which contain harmful chemicals. And for instance, when you pyrolyze materials which contain a lot of chlorine, and then one of the problems is that you also generate HCl in your pyrolysis unit. And HCl is, of course, an issue when you think about corrosion. So in general, you can pyrolyze a lot, many possibilities. You break it down in all kinds of small fragments, but you have to be a bit careful with the formation of particularly the components formed from non-oxygen, hydrogen, and carbon input in your system.
Okay, the next question from wait. Oh, here.
Yeah.
From Maria Lucia Dwi Gama. The question is, what is the advantage of pyrolysis method compared to low temperature method for in bioethanol fermentation? For example, bioethanol fermentation. And the second question, does polymers from pyrolysis BTX would also be easier to degrade rather than the crude oil counterparts? Thank you.
Thank you. Very interesting questions. What is the advantage of pyrolysis compared to bioethanol? My belief is that there is not one method which is good or bad. What you have to do when you do biomass conversions, you actually have to see for a particular source you have in your hands, what is the best way to give it value? And for some biomass sources, it is pyrolysis. For some of them, it is a low temperature step. So the answer is not easy to give. Then the second, when you look at it from a larger perspective, of course, the winner, which will be the best, it will also depend on price. Just very simple, which of the processes has the lowest price. And if you look to bioethanol, it's not the competitor for pyrolysis is not bioethanol from a sugar-containing feeds like sugarcane or sugar beets. Now you have to compare it then with bioethanol from wood. Because we pyrolyze wood and make pyrolysis oil. If you want to compare it with bioethanol, then you have to compare it with wood to bioethanol. And that's a second generation bioethanol process. And second generation bioethanol is also not yet commercialized on a large scale. And that's also in progress. It's by far more difficult to make bioethanol from a woody source than from sugarcane or sugar beets. So it's a bit a difficult question.
Okay, thank
you.
The
second one,
yes,
is the polymer you make from BTX. So the PET we made from biomass. Is that easier to degrade? The answer is no, because what you make, the compound you make, is from a chemical point of view exactly the same as the one you make from the fossil resources. The only difference is then you make it, when you make it from a fossil resource, the carbon you use is very old. It's all more than 1 million years old. If you make it from biomass, the carbon you introduce in your polymer is of a younger origin. That's the only difference. But the properties of the polymer are exactly the same.
And the last question from Anis Christiani from Lipi. What do you think about pyrolysis of combined feed from palm oil waste, and plastic waste into fuels? Thank you.
Very interesting. I told you already, pyrolysis of biomass and catalytic pyrolysis of biomass is very hot at the moment. And also the conversion of plastic waste using pyrolysis to useful products is also a very hot topic at the moment. And indeed, there are quite some studies ongoing where you combine the two worlds, where you actually combine pyrolysis of both waste products and plastics. And so biomass waste products and plastics. And you may say, well, is there an advantage? Why don't you do it separately? And there are indications that you may end up in some synergistic effects. And actually that the yield of the pyrolysis process from the biomass part will be increased when you have plastic present. And that has to do with the fact that when you have plastic present, the composition is different, the CH ratio is different, and the idea is that it has a positive effect on the pyrolysis product you get. It's the case with pyrolysis, and it's also the case with catalytic pyrolysis. So I think there is indeed future to have a good look at the combination of two feeds for pyrolysis processes. If you're interested in palm oil residue pyrolysis, then I can give you an example. If you have empty fruit bunches, the company BTZ, about 10 years ago, they had a pilot plant in Malaysia where they used empty fruit bunches as the feed for a pyrolysis process, and they produced their pyrolysis oils on a scale of about 1 ton per hour.
Okay, thank you very much, Prof. Hiris, for answering all the questions from the participants. I think time is up, and now we will move to the next sessions. Okay, thank you very much, Prof. Eric Hiris,
You're welcome.
for
It was my
being
pleasure.
the
Nice
speaker
to meet you.
of this webinar. Thank you very much.
You're welcome.
Okay, the next session will be guided by the host, Abdul Karim. Thank you very much.
Okay, thank you. We are so grateful. Thanks for excellent presentation by our speakers, and thanks to moderator who has lead this session to make it conducive. The next event, closing remarks by Head of Chemistry Department, Faculty of Mathematics and Natural Sciences, Riau University, Dr. Muhdarina. The time is yours.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. I am Dr. Muhdarina, as the head of Chemistry Department, Faculty of Mathematics and Natural Sciences, Riau University. First of all, I would like to thank all of the speakers, Prof. Dr. Taifo Mahmud from Oregon State University, USA, Muhammad Asif Khan, PhD from Perdana University, Malaysia, Dr. Rehana Begum from Energy Research Institute, NTU, Nanyang Technological University, Singapore, and Prof. Dr. Eric Hiris from University of Groningen, Netherlands, for their willingness to share the highly valuable knowledge and insight to all of us. It is a honor to have all of you in this event. Okay. I also would like to thank all of the participants. In total of 1,243 participants from 10 countries. That's USA, Kenya, Estonia, India, Philippines, Thailand, Taiwan, Malaysia, Japan, and also Indonesia. From the total participants, that are 65% for 2 session participants, 26% session 1 only, and 13% for session 2 only. It is very high pleasure to have you, although we could only meet virtually due to the pandemic. Lastly, I want to thank all of the committees, Ririn Novianti et al., who have strived so hard and given their best so that this webinar could be done nicely. On behalf of the Chemistry Department, Faculty of Mathematics and Natural Sciences, University of Riau, and all the related parts, we have finally reached the end of this event, and hopefully we could meet again in the further event. I sincerely hope all of us always in the healthy condition. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much for closing remarks by our head of chemistry department. The last but not least, the photo session. We will take a picture for a moment. So for all participants, please turn on your video because the committee will screenshot the screens. Okay, in my command, keep smiling. 3, 2, 1. Okay, once again, 3, 2, 1. Okay, I reminded to all participants we need your advice and suggestion regarding this webinar. So you will fill the form that we have sent on chat box. And for access this certificate, you can check your email or you can access this link that you see on your chat box. Okay, finally, we arrive at the end of this webinar. Such as the series of international webinar Green Approaches in Medicine, Chemicals, and Energy. Thanks for joining our webinar. I'm Muhammad Abdul Karim as a second host for today. Forgive for all my mistakes. Keep healthy, goodbye, and see you next.
See you.
Thank you.